Okay, it is on. Huh? Close the door. So, very good evening to everyone. Am I audible? I hope. If anyone there, please tell audio is okay or not. So, am I audible? First, who are online, put your response. I think on YouTube it came and in Zoom it is audible. It is all right? Okay, ji. So I hope you did your paper as according to caliber and capability, and I hope you did it well. So as for easy and difficulty level doesn't matter more. If any paper is easy, it is easy for all. It is difficult for all. So don't worry about that all, right? So it is a competition. And in competitive examination, it is relative, no any like that much marks required. So don't worry about all these things. If you anywhere find it is difficult, like some people are telling physics is difficult, so based on that, don't demoralize yourself, don't going to feel panic. If it is difficult for all. If it is easy for all. Okay. So we're starting with the paper discussion, right? So here it is botany, zoology, both is there. So I'm just going for question number 92. Question number 92 here, it is a botany topics. So we are going to discuss about these 92. Just a minute. I hope you did it correctly. It is an easy question. Nothing to worry in this question. They are telling about the blue and white selectable marker have been developed which differentiate recombinant colony from non-recombinant colony. All you know better. Recombinant colony are white, no color is coming, and non-recombinant colonies are what basically? Blue. Because as you know, beta galactosidase is remain active in the case of non-recombinant, and due to insertional inactivation in recombinant, beta galactosidase will not produce, and due to chromogenic substances when it will react, no color will come in the case of recombinant. So here we are just going with the statement one. Blue colored colony have DNA inserted in the plasmid and they are identify recombinant and recombinant colony. Second one is what? The colonies without blue color have DNA insert. Of course, what the correct answer? Third one is the correct answer. First statement is incorrect. Second statement is correct. Why first statement is incorrect? Because they are telling blue colored have a DNA of insert. Means recombinant blue color, no. Non-recombinant blue color, recombinant colorless. So statement 1 is incorrect and statement 2 is correct in the case of this particular questions. Right, now we are going to deal about the next question. So 92, I'm going to deal about 92 here. Correct key is which one? Statement 1 wrong, statement 2 correct. Just look according to your key. Got the idea? Because the question number varying from key to key, we are going to discuss here only one key. Right. And it is paper code, it is probably written here T3, right? Now we are moving ahead. Next question. After 92, which one is your botany? Because botany and zoology are mixed, na? So these are the some of the issue came. After 92, 93 is also a botany questions in this one, right? So 93. Given below are two statements. We are just reading the statement. Cell of the tapetum possess dense cytoplasm and generally have more than one nucleus. Nothing problem in the statement. It is a correct statement, right? I hope you are also agreeing. It is a correct statement. Reason: presence of more than one nucleus in the tapetum increases the efficiency of nourishing the developing microspore mother cell. It is also correct statement. Got the idea, right? So, and it is why it have more than one nucleus, it is explaining it also. So what will come here? It is going for a correct explanation. So both A and R is true and R is the correct explanation. Got the idea? After 93, we are going to deal about the 94th question. So 94th question is a match of the following type of question. I hope you did it well. So if we are going to match, you just see about pteridophyta, who is there? Salvinia is there. Bryophyta, who is there? Polytrichum is there. Angiosperm, who is there? Salvia is there. Gymnosperm, who is there? Ginkgo is there. So who is coming according to that 94? First is looking as a correct option. 1 have 4, B have 3, Angiosperm salvia, Gymnosperm here ginkgo, D2. So here correct answer is first. 94 it is done. Now we are going to talk about question number 96. 96, have a look here. I hope everyone did it correct because direct line of NCERT. Many times we also asked in our paper, right? So who proposed that genetic code for amino acids should be made up of 3 nucleotides? It is George Gamow. 96 direct factual questions is there. So nothing is there more to discuss about this question. I hope you are able to understand it. So George Gamow is the correct answer, right? Now we are going to deal about the next question. Now we are going to talk about 98 is another botany side question. So have a look here. I think here also you did it well, nothing a problem. So 98 is saying about which of the following is an example of a zygomorphic flower? Family Fabaceae. Who is the member of family Fabaceae? P here. Got the idea? Right? So correct answer is second, 98. Okay, G? Right? After 98, where is the botany? Let me check. 101. 101 is the another botany question. Here it is only in the same page. So 101. So if we talk about 101, protein part of an enzyme is called, very easy line of the textbook. It is what basically? Apoenzyme. Protein part is called apoenzyme. I am not going to teach here right now. I am just going to explaining, simply telling about the answer. Where it is required to explain, I will explain it, right? So apoenzyme, right? It is the correct answer. Now we are going to talk about the next one who are from the botany. After 101, we are going to discuss about the 105. 105 is the next question. So here the question is, which of the following enzyme are not essential for gene cloning? It's a good question, but not a difficult question. So as you better know, restriction enzyme will require. Okay, Ji? Otherwise, how we cut it? The desired gene and vector DNA both. Right? DNA ligase also require in the joining. Okay? DNA mutage, I hope you not heard even. It is given an enzyme name. And DNA polymerase also required because cloning, making multiple copy of a gene. So how the multiple copy is possible? When DNA polymerase is there, then only one DNA to two DNA will going to form. So here they are asking not essential. So who is not essential? Who is not essential here? DNA recombinase is not essential. And who is not essential? DNA mutase is not essential. So what is the correct answer for this question is C and D. C and D is the correct answer of this question. Now we are going to talk about after 105 in botany side, 107 is there. 107 is there. Here in the same page is it? Same page. Actually, zoology, botany are mixed. That's the reason we are facing here a problem. Who is from zoology, who is from botany? So this is also a botany question. Which of the following is a characteristic feature of the gymnosperm? Gymno, definition they ask. Gymno means naked seed plant. So what the answer of 107 will go first? Got the idea? This is the characteristic feature of the gymnosperm. They are the naked seed plant. So nothing more required to discuss here. 109. In Bryophyta, gamma help in. Obviously, asexual reproduction. Gamma cup. Got the idea? Have gamma, they reproduce asexually and forming the new plant body. Got the idea? Right? Not the sexual reproduction. Two are of course eliminatable. You confuse in asexual and sexual. It is a method of which one? Asexual reproduction. Not in the sexual reproduction it involved. So 109 is also over. After that 110 we are going to discuss. Have a look. So 109 answer is A. Got the idea? That is asexual reproduction. After that, we are going to talk about 110. So how many meiotic and mitotic division need to occur for the development of mature female gametophyte from the megaspore mother cell? As I told you, these megaspore mother cell will go in meiosis and forming 4 megaspore. Out of that, 3 usually degenerate. So here how many meiosis occur? 1 meiosis occur. Got the idea? 1 meiosis occur. Got the idea? They are telling female gametophyte formation. So this is going for female gametophyte formation. This is functional megaspore. And these functional megaspore divide how many times? 3 times free nuclear mitosis. We discussed many times this. It is going to form how much? Total 8 nucleus. Out of that, what happened? 3 remain toward the micropylar end, 3 toward chalazial end, and 2 here. It is 7 cell, 8 nucleus structure, and it is female gametophyte. So how many mitosis require? 1, 2, 3. How many meiosis require? 1. So what the correct answer will come? 1 meiosis and 3 mitosis. You got the idea? 110 is also done. Now after 110, who is from the botany side? Let me check. 112 is also there. 112 is also there from the botany. From PGR, this is the question. Got the idea? I hope you did it correctly. Right? So we are going to talk about 112. Read the following statement on the plant growth and development. Partheno can be induced by auxin. It is written in NCERT, parthenocorpine. Tomato. Got the idea? Plant growth regulator can involve in promotion as well as inhibition. It may be promoter, it may be inhibitor. Both type of properties there. Like abscisic acid is inhibitor, auxin is a promoter. An example. Differentiation is a prerequisite of redifferentiation. Obviously. So you know now, first undifferentiated, converting into differentiated, then converted into de-differentiated, and then converted into redifferentiated. So if de-differentiation not occur, how redifferentiation occur? Are you getting the idea? So it is also a correct statement. So if B is also a correct statement. D, abscisic acid is a plant growth promoter. No, it is an inhibitor. An apical dominant promote the growth of lateral word. No. Got the idea? Apical dominant going to apical word. Apical dominance, what basically? apical word, right? Not the lateral word. So it is also a wrong statement. So they are telling choose the option who have correct statement. So A, B, and C. A, B, and C. Who is the option? 112. Fourth option is there. 112 here. Fourth option is there. Which one is correct? Which is incorrect? I already told here. Now we are going to discuss the next. So here, have a look. Oh. Next question is after 112, 116 is there. 116, 116, 116. Yeah, have a look. So if we talk about 116, what we will find? Silencing of a specific mRNA is possible via RNAi because of what happened in RNAi? Complementary double-stranded RNA is forming, right now? Complementary double-stranded RNA is forming. So this is the mRNA whose silencing we want. These are complementary DS mRNA. 5 prime to 3 prime, 3 prime to 5 prime. It is what basically sense strand, it is anti-sense strand. These anti-sense strand, they are going to digest it, and these anti-sense strand pair with this and going to silencing this mRNA to form the protein. So what the correct answer will come? 116, 4 will come. Got the idea? Done. 117 is an easy question. I hope everybody did it. So if we talk about adenylic acid, it is a nucleotide. Adenylic acid is a nucleotide. Adenosine is a nucleoside. Adenine is a nitrogenous base. Alanine is an amino acid. So 117, who is matching? 2 is matching. 1 match with 3, B match with 2, C match with 1, D match with 4. So 117, right? Now we are moving on the next question. After 117, what is the botany size? Let me take 117, 18. No, wait a minute. Now probably 121 is there. 121 is the botany questions. Wait a minute. 121. Right. (121). So it is a certain region question, and it's depend upon the person, but we are finding here, they are telling primary function of the Golgi apparatus is to package the material made by the endoplasmic reticulum and deliver it to intercellular target and outside. Reason is what? Vesicle containing material made by the endoplasmic reticulum fuse the cis phase and Golgi apparatus and then modified and released from the trans phase. Got the idea? So here, 2 possibilities there. People may depend upon here paper setters. Got the idea? But here we are considering it is a correct explanation. Both A and R are true, and it is a correct explanations of the. So it is somehow explaining, right? So while answering, we debated it on also, but we finally make a conclusion. It is a correct explanation. Got the idea? So it is somehow explaining ki from ER it is going to Golgi body. Then here it is also saying the same thing repeatedly. So correct explanation, but very clear-cut correct explanation is not reflecting here. So here it may be a dot, point, a small question mark. Mostly we are finding they will give also the correct explanation, but finally depend upon the paper setter, what were their thought of mind. But here, meanwhile, we are thinking like it is what? It is a correct explanation. Got the idea? Now, after this, we are going to deal about the next question. After 121, we dealt now. After 121, we are going to discuss about 124. Just have a look. 124. What is the main function of the spindle fiber during the mitosis? This is the 124th question number is there. So obviously spindle fiber, what the spindle fiber is doing, they are helping the chromatid to separate, sister chromatid to separate. So what that things will come here? To separate the chromosome. 124, this will be the correct answer. Nothing related to this, nothing related to this. So what happened? Fourth is the answer, right? Which of the following statement is correct about Rubisco? Rubisco has higher affinity for oxygen. It is a wrong answer. It have a higher affinity for carbon dioxide. It is an enzyme involved in the photolysis of water. No. Photolysis of water, oxygen evolving complex is evolving. It is an incorrect statement. It catalyzes the carboxylation of RUBP. It is a correct statement. It is active only in the dark. It is also incorrect statement. So who is the correct statement is there? So third, 125, third is the correct statement. Now we are going to deal about the next. Right. So next botany is which one? 126 is also the botany. Wait. 126 also the botany one. Have a look. So statement, the DNA fragment extracted from gel electrophoresis can be used in construction of recombinant DNA. It is correct. Smaller size DNA fragment are observed near the anode while larger fragment are found near the cathode. So both are the correct statement here. So which option is both the correct statement are there? Fourth option is both the correct statement are there. Got the idea? Right. So both the statement are correct and probability is the line of NCERT. Okay. Now we are moving ahead on the next question. Next question is 127, right? So 127 they are asking which factor is important for termination. So sigma factor for initiation, rho factor for termination. So correct answer is 2. 127 correct answer is 2. Now we are going to talk about the 129, another botany questions. Let me scroll it up. 129. 129 is another botany questions. Have a look here. Histone are enriched in, you better know, lysine and arginine. Right? Direct line of the textbook, lysine and arginine. Histone are enriched with what? Histone and R. Now we are going to talk about the 130. This is also the botany and one of the very easy question from genetic side. Right? So 130. What 130 they are telling? Ki gene R and Y follow independent assortment. They are telling not linkage is not there, independent assortment is there. If pure line capital R, capital Y cross with a small r, small y, F2 generation they are asking. Dihybrid F2 generation, what ratio will come? 9:3:2, 3:1 ratio is coming. Nothing easy question like this, na? Got the idea? So here 130 is also done. Okay, G. Now we are moving ahead. Have a look here. After 130, which one is a botany? Let me check. (133) zoology people are claiming, so we are not going to discuss. 135 is there. 135 is there. 135. Right? So 135 they are telling which of the following microbe not involved in preparation of household product. So in household product you are using lactobacillus for making curd. You are using which one? Yeast. Getting the things or not? Bread making, right? Na? Saccharomyces cerevisiae in bread making. Right. Now we are using Propionobacterium saramani in cheese making. Got the idea? So what we are not using in household, Aspergillus niger, and we are also not using which one? Trichoderma polyesporum. Got the idea? Right? These three are used in household product. Got the idea? Right? So 135, who is not used? A is not used, and who is not used? C is not used. Right? So Trichoderma polyesporum, it is producing which one? Better know, na? Trichoderma polyesporum, what it is producing? Pin your answer. Make somehow like the teaching class. Tell me, Trichoderma polyesporum. No one? All are tired. Answer is coming. 5 are there major, like one is Monascus purpureus, another is Trichoderma polyesporum, another is a streptokinase. Got the idea? So streptokinase is what basically? Clot buster. Trichoderma polyesporums used in which one? Immunosuppressive agent, right? And Monascus purpureus, what basically? Blood cholesterol lowering agent. Okay, anyhow, now we are going to talk about which question? Question number 136. One student is telling which one? Botany? Some written botany. 123 is also botany. 123 I left. Let me check. Give me a moment. 123 I left here. I left 123. Thank you very much. 123 we are also going to discuss. You just simply make which one? Apiblast is a rudimentary cotyledon. Perisperm persistent nucellus. Right. Scutellum is present in which one? Cotyledon of monocot. Non-albuminous seed, what basically? 123. You got the matching done? So what will coming? C is matching with which one? C is, what I did? Rudimentary cotyledon. C with 4. Where C with 4 is there? Only this option is there, na? So 123 it is 4th. C with 4. Apiblast is rudimentary cotyledon. This will going to help to eliminate all. Got the idea? 123 also done. Now we are going to move ahead. So we reach up to which one? We already discussed about 130, 133. That is 135 discussed. So who is not using household product? One is Aspergillus niger and another is which one? Trichoderma polyesporum. Now 136. We are going to talk about 136. They are using foam breaker. Diagram based question. So who will come? C is the foam breaker here. Here C is what basically? Foam breaker. These are help in agitation. When due to agitation foam will form, this C will break. Got the idea? Now we are going to deal the next question, that is 137. Have a look. 137 is also botany. The name of class, NCERT based question directly. What happened? Transfer of group occur. What the idea? So which enzyme will use here? Transferase. NCERT line is there. Simply they ask. Got the idea? So transferase is the enzyme. Okay, 137 is also done. Now after 137 we are going to move on which one? The next one, 138. So 138 is also botany questions. Have a look here. So 138, have a look. So chlorophyll A, what is the color? Blue green. Chlorophyll B is which one? Yellow green. Carotenoid. So these are going to say now A is 3. A is 3, B is 1. A is 3, B is 1. Who is coming? Right. C become xanthophyll. Blue green, carotenoid, yellow to yellow green. Got the idea? Right. So 138 is also done. Now we are going to talk about 139. It is a good question. Listen it carefully. 139 is a good question. It is somehow tricky also and good also. 139 we are going to discuss. So they are asking the correct sequence of event in the life cycle of bryophyte. So life cycle of bryophyta, we will start with haplontic, because main plant body is which one? Gametophyte. Main plant body is gametophyte. So first we will talk about attachment of gametophyte to the substratum. Then we will talk about release of anthrozoid into the water. Then what happened? Fusion of anthrozoid with the egg. Got the idea? Later on what happened? Reductional division occur in the haploid spore, and then finally what happened? Formation of the sporophytic plant body occurs. So what the correct answer will come? Second will B, E, A, D, C. Got the idea? So first gametophyte is attaching to the substratum, then What happening? Release of anthrozoite into the water. After release of anthrozoite, what happened? Fusion of anthrozoite with the egg will happen. After that, what happened? Formation of a, after that, what happened? D, formation of a sporophyte happen. And then what happened? Just a minute. Huh? Let me work out on it. Just a minute. Huh. So 139. It is the very first event. What is the very first event? Attachment of gametophyte is the first event. Got the idea? Second event is what? Basically release of anthrozoite is the second event. Got the idea? The third event is what? Basically C. The third event is which one? Fusion of anthrozoite. After release in the water, it is fusing here. It is the third event. Fourth event, what happened here? Fourth event, what happened here? Formation of a sporophyte and reductional division and wait, what I'm doing. So first attachment, second release of anthrozoite into the water, then fusion of anthrozoite, third event. Now B, E, A, up to here no problem. Now here D and C. D, formation of a sporophyte. And then what happened? When a sporophyte will form reductional division to produce haploid spore. So who will come? Second as the answer, right? Second as the answer. Now we are going to talk about 140. Sequence based question 140. 140, 140, 140. So here what is the answer coming? Second coming, finally. First what happened? Formation of a sporophyte occur. When a sporophyte occur, then what happened? Haploid spore will produce. Reductional division to produce haploid. First, a sporophyte will form. A sporophyte is 2N. Then what happened? Meiosis occur, and after meiosis, who will form? Haploid spore will form. So nothing a problem here, right? So who will come first? D will come first, and then who will come? C will come. Got the idea? So B, E, A, D, C. It is the second is the 39 correct key. Got the idea? Now after that, we are going to deal about 140. So 140, 140, 140, wait a minute. Here it is a easy questions. So you can say about Christie present in which one? Mitochondria. Right? Cell membrane made up of phospholipid bilayer. Cilia have role in cell movement. And centromere have role in cell division. Match it 140. What is the correct match in the 140? 3 is there. 3 is there. A2, B3, B3, C1 and D4. D4. So up to 140 question I discuss. Now what the sequence will come now? Zoology people will come and discuss up to 140. After that we will go for another remaining part of the botany, right? And then another zoology people will come and finalize this. Thank you very much.
भाई बंद कर लो उनको दे दो
90
We cannot just take mic. You can just take mic. In your hand, you can take mic.
If you hold it,
Let him.
Okay, very good evening. We can start with the question number 91. I think 91 question number is the 91 is the zoology question. The team members can say the question number. I think 91 zoology kind of.
140.
91, right? It's an easy question. The hormones and so progesterone, it goes with the corpus luteum, and in fact, relaxin can also go with the corpus luteum, but we don't have any other option going like that. So progesterone goes with corpus luteum, and give me the question. Yeah, 91, 46, 46th one, right. Okay, so progesterone with corpus luteum, and then relaxin goes with ovary, right? The C melanocyte stimulating hormone goes with pars intermedia. It is melanocyte stimulating hormone, full one. And catecholamines with adrenal medulla. It is adrenal medulla. Answer is fourth. The next question, zoology question. I hope I am audible to everyone. So next zoology question is 95th one. Again a match the column question, 95th. 95th one, right? So again an easy one, very easy one. So the hormones, the heart hormone is ANF, atrial natriuretic factor. The kidney produce erythropoietin, and GIT produce secretin, and adrenal cortex, it produces aldosterone. So here we can match it, and we can go with the answer third one. 95th answer goes 3, right? Now next question, next zoology question is the 97th one. 97th. Which of the following is the unit of productivity of an ecosystem? Now, productivity means per unit area, per unit time. And per unit time is going in third one, which is kilocalories per meter square per year. So, answer of the 97th one is the third one. So, next geology question is the evil quartet 99, before 99th. Okay, directly 99th one. The evil quartet goes with causes of biodiversity loss. Ex situ conservation is cryopreservation. Lantana camara is this one. Alien species invasion, and dodo is the recently extinction. So here, 99th one, the answer is the first one, right? Now the next zoology question is the 100th one, 100th, okay. So 100th question is down here. Audibility is there. I think we have not seen the. Am I audible? Any one of you can just say okay. Or thumbs up if audible is there. Audible? Okay, good. So then next one is the 100th question. 100th question is in ecosystem there is a unidirectional flow of energy from sun to producer to consumer. It is correct. And ecosystem are exempted, whereas ecosystems are not exempted from the second law of thermodynamics. We know that both first and second law are applicable. So one is correct and second one is wrong. If statement 1 is correct and statement 2 is incorrect. So here this one is answer is the second one. Statement 1 is correct, but the statement 2 is incorrect. Next is after 102. 102. After the 140th, we'll go one by one. So this is, what is happening. Because of this, we are taking extra time. Twins are born to a family. Now, these are one boy, one girl. My previous faculty can say this answer much better than me, because so this is like one boy, one girl. They both are born at the same time. That means they are not the monozygotic one. They are the fraternal twins. So 102 is answer is 1. Next question is 102. After 102 we have 104. 104 is this one. In frog, the renal portal system. The renal portal system is lower part of the body and the kidney. The renal portal vein goes through the kidney. So answer of 104 is 3, right? Then next one, after 104 is 106. And this is the one which is real. People have different opinion in this one, and even the institutes have given different answers. But I can only say whatever I know about this one. This is, see this one. This mother is affected. Females are carrier. Both the daughters got the gene. In this one, you can see all the carriers are females only. Male is affected, but none of them, you can see this one, all females are carrier. And this means that this is not a dominant one. Carrier think only there in the recessive one. Now, if you see that, if we consider this as autosomal, autosomal क्या होगा बेटा? Autosomal will be small a affected. It is recessive, pakka sure. It cannot be dominant because it is a skipping of generation. It is recessive. Recessive pakka. That is step one clear. Now, recessive means small a affected. Small a affected, right? Now, small a affected, whereas this is a carrier. A dot means it is a carrier. Capital A, small a, and capital A, capital A. It's a normal one, right? So, which means that this cannot come. This small a cannot come because the father does not have the small a, right? Which means that if we consider this as X-linked recessive, this is going. So this is an X-linked recessive considering the females are the carriers one. Now, X-linked recessive, you can use the Punnett square method. I always say the Punnett square method, which means the father one is normal. Father is normal. Punnett square. Father is affected one, and the mother is a carrier. Father is affected, mother is a carrier. Okay. So now we can see this one. This is a carrier. This one is unaffected boy, and this one is normal boy. So out of these four children, you will find that two of them are the one which are carrier. Okay, two of them are carrier. The daughters are carrier in this case, right? Because you can see the father is affected and mother is a carrier. So that means both the daughters are carrier. Okay. Right, we are going, mother is a carrier. I have to just quickly take this once again. Father is affected. Capital X, got that? Father is affected, right? Mother is a carrier. No, this is a carrier daughter. Affected daughter. This is a carrier daughter. This is affected son, and this is a normal son, which means that 1/4 out of 4, 1 is a carrier. Make sense? 1 is a carrier. Let's see the options, and accordingly we can tick the option. So here 1/2, 1/8, 0, and 1/4. We can go with the 1/4. Make sense? 1/4. So answer to question number, pedigree question is 1/4. Option 4. Okay. So these are the questions where some calculation required. In all other question after 106, we can go with the next one. 106. After that we have 108. 108, a very easy one. The first menstruation is menarche. And the last one is menopause. We all know that. 111, 111, right? Echinoderms, 111. Before 111, any question in zoology? No. 111, right. Role of water vascular system. Respiration and locomotion, excretion and locomotion. So respiration and locomotion, we can go with this one, and then we can go with the capture and transport of food. So we can say respiration and locomotion, A and C. So A and C goes with A, all right? 111. 111, answer is 1. After 111 we have 113. After 111 we have 113. Which of the following type of immunity is present at the time of birth? This is innate immunity, right? Innate means inborn. Innate immunity. Why? So answer is 113. Answer is 1. Now 114 is the zoology one. So we can see why can't insulin be given orally to diabetic patient? Now, insulin is a protein, and like any other protein, it can be digested in the stomach, right? Or in the small intestine. So, it will be digested in the gastrointestinal tract. Direct answer, 114, answer is 1. After 114, if my team can tell me the next question, zoology 115. Now, this one is the Verhulst-Pearl equation. That means logistic growth model. Logistic one, dn by dt is equal to Rn, k minus n upon k. Which one is going like that? So option number 1 is going dn by dt is equal to Rn, k minus n upon k is the correct answer, right? Now next one, 116 is not the one, not the zoology one. 118. Frogs respire in water by skin and buccal cavity. The statement is wrong, not by buccal cavity, only by skin and on land, by skin, buccal cavity and lungs. So it is true for land but not true for water. The statement is true for the statement is false for water but true for land. 118 answer is 2. All living members of the class Cyclostomata, they are the ectoparasites on large fishes. We know that they are ectoparasites on large fishes. Now the next one after 119 is identify the 120. Okay. After that 120th question, 120th is this one. Identify the statement that is not correct. The heavy and light chain are held together by disulfide bonds. The correct one. Antigen binding site is located at the C terminal. Now, constant terminal is not the one where the antigen binding site is present at the. This is the antigen binding site, the F fragment AB. The C terminal is a constant one, FC. So this is a constant in all the antibody. The variable one is this one where the antigen binds, not the constant one. So the correct answer to this one is 120. We have to find the incorrect in this one. Antigen binding site is located in the C terminal is wrong. So 120 answer is second one, 2. 120 answer is 2. Then next one is 122. 122. The reductive division for human female gametogenesis start earlier than that of the male gametogenesis. That is absolutely true because it starts in the embryonic life. In the fetal life it begins. So it is obviously correct. The gap between the first meiotic division and the second meiotic division is much shorter for males because it is not interrupted in the males. In the females it is interrupted, right? It is interrupted 2 times, one before that division and one after the division. So between first and second there is a big gap. So these A and B are correct. The first polar body is associated with the formation of the primary. This is wrong because it should be secondary oocyte. Luteinizing hormone surge, LH surge, leads to disintegration of the endometrium. No, LH surge leads to disintegration of the Graafian follicle, leading to ovulation. So statement A and B seems to be correct. So answer A and B are correct. Option number 4 is the correct one of question number 122. After 122, this seems to be a botany one. 123, 124, 128, 128 one. We'll just do this one. It's an easy question. Who is Chacha? वाह, पेपर ने बुरा हाल कर दिया है चाचा. वाह, very good. So this is the adrenal medullary hormone means catecholamines. Catecholamine, right? Adrenaline. It causes pupillary constriction. Wrong. Option A is wrong because it causes pupillary dilation. So A is wrong. It is a hyperglycemic hormone. It is correct because it causes glycogenolysis. It is hyperglycemic. It is correct. B is correct. It causes piloerection. Yes, it causes goosebump. Piloerection is correct. And the fourth, that is D1, is it increases strength of the heart contraction. Absolutely correct. Sympathetic stimulation causes the stimulation of the ventricular contractility. So B, C and D are correct. A is wrong. Let's see which one is matching. The first one is matching. 128, first one is matching. So next one after 128, next one is 131. 131. So after that 131. Which of the following hormones released from the pituitary is actually synthesized in the hypothalamus? We know that the two hormones synthesized in the hypothalamus are ADH and oxytocin. One of them is given here, that is ADH. So answer is first one only. It is ADH, right? Now after this one, we have the 132. And the first assertion reason which I have seen here in today's paper about zoology is this one only. And this is the one which we have done so many times. Assertion all vertebrates are chordates, but all chordates are not vertebrates, because yes, some chordates are urochordates as well as cephalochordates, and they are not vertebrates. Okay. So now they see the reason what they're saying: the members of this subphylum Vertebrata possess notochord during the embryonic period. The notochord is replaced by the cartilaginous or bony vertebral column in the adult. Yes, because the notochord is present only in the vertebrate. And they are so that means they are not all are vertebrate. The vertebrata, the subphylum Vertebrata are vertebrates, means urochordates, cephalochordates. These two chordates are invertebrate chordates. So this reason is correct. The reason is correct. Assertion and reason both are correct, and the reason explains the assertion. So here answer going with 1. A and R are correct, and our answer of question number 132. Assertion, reason both are true, and R is the correct explanation of A. Answer is 4. 132 answer is 4. Now 133. Fig fruit is a non-vegetarian fruit as it has enclosed fig wasp in it. My gosh. Now this is what shall I say? Now, you know that in this fig and wasp mutualism, what happens that the female wasp dies inside the fruit, right? And then the eggs hatch, male and female, they both are born, and they both, males and female, they both copulate, and the male, they make way for the females, their sisters to come out, their pregnant sisters to come out. The male make a randram in the fruit, and from those holes the females come out and they fly and they go. In this meantime, they just dust themselves with the pollen. So that's for cross pollination. Now see, the female has died inside the fruit. So in a way, it is, but no, in the question what they have given is fig fruit is a non-vegetarian fruit as it has enclosed fig wasp in it. No. Now this is incorrect, because what happens that the enzymes of the fig, they digest this female wasp. So you don't get a wasp inside. So right, it is not vegan, we can say. It is vegetarian but not vegan. So those who are vegan, they should avoid the fig. However, the fig which are available in the market are the one which developed by parthenocarpy. So we are talking about some of the species which are fig and these wasp pollinators, mutualism, right? Okay, but so here we can say this question. Statement 1 is incorrect, because you don't get the fig, the female inside the fig, it is digested by the enzymes. And statement 2 is correct, because fig wasp and fig tree exhibit mutual relationship as fig wasp completes its life cycle in fig fruit, and fig fruit gets pollinated by its fig wasp. They are mutualist, they are tight mutualism can be seen. Okay, beta? So here, statement 1 wrong, statement 2 correct. Let me see which one is going like that. Statement 1 is incorrect, but statement 2 is correct. Option is 3. Option 3 is correct. Then sweet potato and potato. Yes, these are analogous, and analogous, what they indicate? They indicate convergent evolution. Analogous structures indicate convergent evolution. So answer of 134, analogy and convergent. That means option number 4 is going like that. 135 is not zoology. Next one.
134 is over.
134 is analogy and convergent. Option number 4. Answer 4. So this is up to 140. Now we will. I think we are not okay with this system, like first botany, then zoology. Now from here onwards, botany, zoology, alternatively we'll be doing. I think that is better for you guys, okay? So now to the botany team, I am transferring, okay.
From here, this one, which is yours.
Good evening, everyone. So I am Sunita. I'll continue from 141 question. I'll continue with the zoology. Sir, Ajay sir.
Hello all.
Ajay sir, he'll continue botany questions. From what 140, we'll parallelly will do it. Okay, it will be easier for you to mark. Okay, so 141 zoology question from human reproduction, and yeah, this is the question. In human pregnancy, the major organ systems are formed at the end of 12 weeks. Yes, it is correct. And B statement says that in human pregnancy, the major organ systems are formed at the end of 8 weeks. So this is wrong because it is formed completely by the end of the 12 weeks. Then this is wrong. And C statement says in human pregnancy, heart is formed after 1 month of gestation. This is right, like direct line of NCRT only. In human pregnancy, limbs and digits develop by the end of 2nd month. Okay, this is also right. In human pregnancy, the appearance of hair is usually observed in the 5th month. So we consider this also right. So with this answer is A, C, D, E. Okay, so option 3 we have to mark. So 141 will go with option 3. Next,
142 is botany. टाइम लगेगा चलो देखते हैं 142. This is a kingdom question, 5 kingdom classification question. Fine. So you see, we will start with from lower to higher. We have to go for a respective kingdom, arranged from the increasing order. So we will first of all start with the prokaryotes. So see, we have only first prokaryote, one prokaryote, C it will come. Then you will come unicellular eukaryotes. So unicellular eukaryotes, eukaryotic or with cellular body organization. So this will be E. Okay. Then we will come to fungi, multicellular heterotrophs with cell wall made up of chitin. So A. Now Plantae and Animalia. So first we have to keep Plantae. So eukaryotic autotrophs with tissue organ level of body organization. So that will be D. And Animalia last, heterotrophs with tissue. So this will be B. CEADB. So CEADB, our first option is the correct answer. Okay. First option is the correct answer. CEADB. Okay. 143.
Uh, 143. See here, which are correct? Here, this question is from human health and diseases. First statement is about computed tomography and magnetic resonance imaging detect cancers of internal organs. This is right. Okay. And next line is chemotherapeutic drugs are used to kill non-cancerous cells, it seems. But actually, the drugs which we use, they destroy cancer cells only. So this is wrong. So A is right, B is wrong, and C. Alpha interferon activates the cancer patient's immune system. Yes, that is right because that is immune modifier. So and helps in destroying the tumor. This is correct. Chemotherapeutic drugs are biological response modifiers. It is given. That's wrong because alpha interferons are actually considered as the biological response modifiers. This is wrong. In case of leukemia, blood cell counts are decreased. In case of leukemia, actually blood cell count will increase. Okay. So with this, we considered this statement is correct, and this statement chemotherapeutic drugs are used to kill cancer cells. So here it's given non-cancer, so wrong. Alpha interferons activate the patient's immune system. This is correct. Chemotherapeutic drugs are biological response modifiers is given. This is wrong. In case of leukemia, blood cell count increases. So this is also considered wrong. So with this answer is A and C only. So let us see where that particular thing is there. A and C. So option 3 is correct. And 144, it's from Biotechnology and Applications chapter. Which of the following genetically engineered organisms was used by Eli Lilly to prepare human insulin? So this we came across under genetically engineered insulin. So from that chapter, and Eli Lilly, American-based company, used Escherichia coli bacterium. So option 4 is correct for this. Okay. Option 4, bacterium, particularly Escherichia coli bacterium is used. Okay, 145 botany.
So you see 145 is a very, very simple question. It is which pattern of inheritance for polygenic trait? Now, if you see Mendelian trait, Mendel said one character is controlled only by one gene. And you know that polygenic character 1, that is the skin color. And genes, non-allelic genes controlling the character 3, A, B, C. It means it is not following the Mendelian pattern. So answer will be non-Mendelian inheritance pattern. A very simple question here. Fine. Now 146. Which of the following are the post-transcriptional events in an eukaryotic cell? So after transcription in eukaryotic cell, first cell is splicing. But the splicing takes place inside the nucleus. Okay, here it is written, transport of pre-mRNA to cytoplasm prior to splicing. That is why it is wrong. It should be after splicing. So this statement is wrong. Removal of introns and joining of exons, absolutely this is correct. This is splicing. Addition of methyl group at 5' and yes, methyl cap, it is added. Addition of adenine residues at 3' of hnRNA, of course, poly A tail is added. Base pairing of 2 complementary RNAs, no. If there will be base pairing of 2 complementary RNAs, there will be no translation at all. Gene silencing will take place. So this is a wrong statement. So correct statements are B, C and D. So B, C and D are the correct statements. Answer 1. Okay, next. Which one of the following phytohormones promotes nutrient mobilization? So as this nutrient mobilization come, you have to think nothing and you go for cytokinin, because delaying of senescence is caused by gibberellin and cytokinin both. But when it comes to nutrient mobilization, you will have to go for cytokinin. So that is the answer is cytokinin. Fine. Next. How? 48. Over to you, ma'am.
148, it's a new question for you. You can find this line in NCERT. Human physiology introduction is there, right? Once you open that particular page, it's a direct NCERT line only, but from introduction of human physiology. Okay, so it's a new question for you. I don't know how many members marked it right. Which one of the following statements refers to reductionist biology? So here, it answer is physico-chemical approach to study and understand living organisms. This is the NCERT direct line. So answer 4 is correct here. It's not about physiological approach. It's not only chemical approach. It's not behavioral approach. It's about physico-chemical approach to study and understand about living organisms. Okay. So option 4 is correct. We followed NCERT exactly for this. Okay. Next question. 149 from human physiology and from first, breathing and exchange of gases. Actually, this year they did not follow exact way. It is for human physiology. Locomotion and movement, neural control and coordination. They did not ask any questions. It's really shocking. And even evolution also weightage has taken very less. Okay, animal tissues also they did not ask any questions. So no issue. And most of the questions are asked from human health and diseases actually. So this question from breathing and exchange of gases, match list 1 with list 2. List 1 says emphysema, angina pectoris, glomerulonephritis, tetany. Okay, now we have to match list statements given from list 2. Emphysema, you all know very well. Answer is damaged alveolar walls and decreased respiratory surface. So for emphysema, we have to opt second statement here from list 2. Angina pectoris, it is about acute chest pain when not enough oxygen is reaching to heart muscle. Okay, so this is a symptom actually before anyone suffers from heart related issues. So B, 2, 3. And glomerulonephritis, you know very well, it's about inflammation of glomeruli of kidney. Tetany, it's about rapid spasms in muscle due to low calcium in body fluids. Okay, so this match the following is made from all chapters of human physiology. Covered all topics here only. So with this option, emphysema to 2, emphysema A2, and B, it has to be 3. B has to be 3, so option 3 is correct here. So next question is, epiphytes that are growing on a mango branch is an example of which of the following? So epiphyte, this is from organism population topic, population interaction concept. So answer is commensalism. Epiphytes means the plant which is growing on another tree. So here commensalism, one is taking the benefit, another one is neutral. So option 4 is correct here. 150, option 4. And for this, I already told you 3, option 4 here. So 149, 3, 154. Next to botany, sir.
Okay. 150 fun is a matching question. So you know Harsha and Chase, they provided unequivocal proof that the genetic material is DNA. Fine. So DNA is the genetic material. So A4. A4 we have 2 options here. Let us see. Then euchromatin, you know euchromatin is the loosely packed chromatin. So B3, A4, B3, we have got our answer already. But let us see once more. Frederick Griffith. Griffith is used the bacterium Streptococcus pneumoniae. So C will be 1. And heterochromatin is the densely packed, to D second. So our answer is 2. Answer is 2. Alfred Harsha and Martha Chase, they proved that the genetic material is DNA. Then euchromatin is the loosely packed and it is genetically active chromatin. Then Griffith performed his transformation experiment on Streptococcus pneumoniae, and heterochromatin is genetically mostly inert and densely packed chromatin, dark staining chromatin. Fine? Okay.
Yes. 152, it's from molecular base of inheritance chapter, HGP concept. Which chromosome in the human genome has the highest number of genes? Its answer is chromosome 1. Chromosome 1 has most number of genes, 2968 genes are present over there. So next about 153. Sorry. 153. What are the potential drawbacks in adoption of the IVF method, in vitro fertilization method? Okay, what are the what side effects or negative drawbacks related to that, they asked. So A is saying high fatality risk to mother, so no problem at all. So we know, I mean, her survival, no issue at all, and so we no need to consider that. And B, expensive instruments and reagents. Yes, it is correct, and it's very costly. Technology is very costly also, and where they have to use all the costly instruments. And husband, C is saying that husband wife necessary for being donors. That's not the drawback at all. And D is less adoption of orphans. Yeah, because their own children they can have, so at that time they don't show interest to go for adoption of orphans. So this one also a drawback only. Next, not available in India. It's available in India, so no need to consider that. Possibility that the early embryo doesn't survive. Yeah, there is a some technical issues will be there where the embryo can can be destroyed in the artificial environment. So we consider here B, D, F. B, D, F. Option 4 is correct here. Okay. Next, we'll move on to question number 154. Yeah, this question is from human reproduction. Here, match the following. You would have felt very comfortable to solve these questions. List 1 is about sperm, parts of the sperm. Okay, head, middle piece, acrosome, tail. List 2 is about the related term, composition. We need to find out. Okay, head, genetic material is there. Then middle piece, as mitochondria, spirally called mitochondria is present, and that supports that's the source of energy. Acrosome, it's filled with all sperm lysins, which are useful to dissolve the secondary oocyte membrane. So we can consider sperm lysins, means group of enzymes we can consider. Tail, yeah, tail definitely useful for sperm motility. So with this option, 4 is correct. A4 and B3 and C1 and tail, for motility. So option 4 is correct. Okay, next.
One 55. See here, the eukaryotic ribosomes are 80S and prokaryotic ribosomes are 70S, correct? Each ribosome has 2 subunits, very correct. The 2 subunits of 80S ribosome are 60S and 40S, while 70S are 50S and 30S, absolutely correct. The 2 subunits of 80S are 60 and 20, no, this is wrong. Wrong, this is 50 and 30 and this is, okay, fine, so this is wrong, okay. The 2 subunits of 80S are 60 and 30, no, they are 60 and 40. And 70 are 50 and 30 is correct, but this is wrong, 60 and 40. So A, B and C are correct. So answer will be A, B, C are true. Fine. Which one of the following is an example of non-distilled alcoholic beverages? I think no one has done this correct answer wrong. Non-distilled alcoholic beverages are less than the 13% alcohol. They are wine and beer. And here your option will be beer because wine is not in the option. Fine? Okay, beer. Next is a zoology question.
– 157. Who is known as the father of ecology in India? So it's a direct line of NCERT. Answer is Ramdev Mishra. Ramdev Mishra. Okay, he is considered as father of ecology in India.
Now 158, a very, very simple question. In seeds of cereals, the outer covering of endosperm. The outer covering of endosperm is proteinous, which is called as the aleurone layer. The rest of the portion in the center is the starchy. So this answer will be aleurone layer, which is the outer proteinous covering. Okay. Next is zoology. Fine.
Yeah. Next question, 159. Which of the following statement is correct about location of the male frog copulatory pad? You all are very familiar with this question. We many times we gave in our papers also. First digit of the forelimb. Okay, it's a temporary structure. Only in the breeding season it appears and helpful for holding the female frog. Okay, during copulation. Okay, so option 3 is correct for this. Next, botany.
(160). A specialized membranous structure in a prokaryotic cell which helps in cell wall formation, DNA replication, respiration. You know, there is an infolding of the cell membrane, which is called as the mesosome, and mesosome is involved in function like the DNA replication, cell wall formation, cell division, all these functions. Also, it is involved in aerobic respiration if the bacteria is aerobic. So many functions. The answer will be mesosome. Fine. Okay, next. Next, transfer RNAs and ribosomal RNA do not interact with mRNA. This is not possible. You know, when the translation has to be initiated, first of all, the mRNA comes and binds to the ribosomal RNA of the RNA, okay, of the ribosome. And then the tRNA will come along with the first amino acid to bind to it. So all mRNA, rRNA and tRNA are involved in the process of translation and they interact with each other. So this is a wrong statement. Fine. Okay. Next. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. Yes, this is correct. So statement 1 is incorrect. Statement 2 is correct. Okay. And RNA interference was discovered in a nematode, Rhabditis elegans, which is a non-pathogenic nematode. So statement 1 is incorrect, but the statement 2 is correct. Fine. So answer for 161 will be 3. 162 is a zoology question.
162. What is the name of the blood vessel that carries deoxygenated blood from the body to the heart in a frog? Okay, this is from frog chapter, circulatory system. So you know it has three chambered heart. Okay, so right atrium, left atrium, and only one ventricle is present. You know very well that on dorsal side of the heart, triangular shaped one chamber, additional chamber is present. That is called this chamber is called as actually. This chamber is called as sinus venosus. Sinus venosus. Okay, this sinus venosus is filled with deoxygenated blood, and two precaval veins and one postcaval vein collects the deoxygenated blood from body parts and pour that into sinus venosus. From there, it goes to right atrium. So vena cava is the right option here. So vena cava, third option is correct for this question. Next question is from botany.
Now, there are two statements here. Let us see first statement, 163. In RNA world, RNA is considered the first genetic material evolved to carry out essential life processes. Yes, it is correct. RNA acts as a genetic material and also as a catalyst for some important biochemical reactions in living system even today. Okay, being reactive, RNA is unstable. So that was the problem. That is why RNA was not considered to be a very good material to function as a genetic material because of its catalytic nature, because it was unstable. So, but statement 1 is correct. And statement 2, DNA evolved from RNA and is a more stable genetic material. It double helical strand being complementary resists change by evolving repair mechanism, because of suppose one of the strands get damaged, since the two strands are complementary, the other strand will function as a template to resynthesize the new damaged strand. So that is a very good mechanism. Fine. So both the statement 1 and statement 2 are correct. Fine. Fourth. Next, I think it is zoology, XC2 conservation.
Which one of the following is an example of ex situ conservation? You know very well that threatened species, critically endangered species are taken out from the natural habitat and given more protection in the artificial environment, like zoo, botanical gardens, wildlife safari, cryopreservation methods, and gene banks, stem cell banks, etc. In that case, zoo and botanical gardens are the examples for ex situ conservation. Okay, like artificial setup. The remaining options are related to in situ conservation, where organisms are given more protection in the natural habitat. Okay, so for 164, option 2 is correct. Okay, next botany question.
165.
Yes.
So 165 is a very simple question. Which one of the following enzymes contains heme as prosthetic group? You know that heme as prosthetic group is found in catalase and peroxidase. So here answer is given as catalase. Okay, so it will be catalase. Next, 165. This is a bit tricky question, we can say, because given below are the stages in the life cycle of pteridophytes. Arrange the following. You see, earlier there was a question on bryophytes. So since bryophytes, the predominant stage is the gametophyte, we'll have to start from the haploid stage of the gametophyte stage. But since pteridophytes, the predominant stage is a diploid sporophyte, so we'll have to start from the sporophyte or the diploid stage. So let us see, the part that is producing the spore will come first. So let us see. Fine. So I think this will be the first one, meiosis in the spore mother cell. Okay, then the spore mother cell will produce the spore, and this spore will produce the prothallus. So prothallus stage. Then in the prothallus there will be formation of archegonia and antheridia in the gametophyte. Okay, prothallus. Okay, formation of transfer of antherozoid. Okay, prothallus. Okay, so prothallus, then formation of archegonia and antheridia. So D, formation of archegonia and antheridia, then transfer of antherozoids to the archegonia in presence of this D, and ultimately there will be fertilization C. So BADEC, BADEC, BADEC. Fourth option is there, BADEC. That will be the correct option, because as I said, we are going for pteridophytes. So we will start with the sporophytic phase, which is the predominant phase in the pteridophytes. Fine. Okay, next. Which of the following organism cannot fix nitrogen? So see, there is only one algae here. All other are cyanobacteria, and you know cyanobacteria, they have nitrogenase enzymes to produce the to fix the atmospheric nitrogen into ammonia. So answer will be only D. Okay, D only. I think next one is
168 is
160.
168. This question is from Animal Kingdom. While trying to find out the characteristic of a newly found animal, a researcher did the histology of adult animal and observed a cavity with the presence of mesodermal tissue towards body wall. Because you know, right, like the fluid-filled cavity present between body wall and the gut wall means ectoderm and endoderm. And if it is lined by mesodermally derived peritoneum lining continuously, then you can call that as a true coelom. But here they are saying that this mesodermal tissue is present towards the body wall, but not there towards the gut wall, it seems. Okay, then it has to be it has to be considered as pseudocoelom as it is discontinuous. So for this, we can mark option 1. It is pseudocoelom. Here mesoderm is arranged as scattered pouches between ectoderm and endoderm, means body wall and gut wall.
Sorry. 169. It is a morphology question, very simple question. Given below are two statements. Statement 1, in a floral formula, this stands for no, this stands for actinomorphic flower. So this is wrong. And G2 below, bar below G stands for inferior ovary. No, this is also wrong. Bar below G stands for superior ovary. So both these informations given are incorrect in this statement. Fine. Next. Statement 2. In a floral formula, this stands for actinomorphic flower and this stands for superior ovary. Okay, so statement 1 is incorrect and statement 2 is correct. So let us see. The answer will be statement 1 is incorrect, but statement 2 is correct. So answer will be 3. Okay. Next, again, it is a zoology question, 170, ecosystem question. So now we'll discuss it. Okay. Fine.
170. Given below are two statements. Statement 1. This question is from ecosystem chapter. Statement 1 is primary source of energy in an ecosystem is solar energy. That's true. That's correct. Statement 2. The rate of production of organic matter during photosynthesis in an ecosystem is called net primary productivity. But actually, net primary productivity is available energy. What to say for the next trophic level? Okay. So GPP minus R is equal to NPP. So this statement is wrong. It's not NPP. It is about gross primary productivity. So I consider this as correct and this as incorrect. So with this option, first statement is correct and next statement is incorrect. So option correct and second option is correct. Option 2 is correct. Okay, so next one, which of the following diagram is correct with regard to the proximal and distal convoluted tubule of the nephron? This is just based on the textbook picture only, tubular reabsorption and tubular secretion concept, very nicely given this time. Okay, and here see option 1 is saying that H plus and H3 in the PCT. This side it's a PCT, this side it is shown about DCT. They had represented with PD like this. Okay, here the up arrow mark is representing the components which are taken up into the bloodstream from the renal tubule, from renal filtrate. So it is like tubular reabsorption, it is like tubular secretion. This arrow mark is representing tubular secretion from blood. Concepts, substances are added to the renal filtrate. Okay, so with this, you can apply the concept on these pictures, and option 1 is correct here. Option 1. Many would have got confused in this question also. And see here, H plus and H3 are secreted, and bicarbonate ions, NaCl, H2O, these are reabsorbed. Coming to DCT, because hormonal regulated part of the nephron, this DCT is. So NaCl due to aldosterone effect, and H2O due to ADH effect, and bicarbonate to balance the pH, it can be reabsorbed, and K plus H plus can be secreted to and added to the renal filtrate. So option 1 is perfectly correct for this question. The remaining, like picture based also, it's not considered.
172, a very simple question. Streptokinase is produced by bacterium streptococcus. Okay, this is called as a clot buster. So it busts or breaks the blood clots. So it is used for removing clots from the blood vessels. So simple question. Microbes in human welfare chapter, from where you get this question. Again, 173 is a zoology question.
Yeah. Question number 173, it's from circulatory system chapter, physiology topic. Then cardiac activities of the heart are regulated by A, nodal tissue, correct. Okay, nodal tissue is the specialized cardiac muscle fiber and that is auto-regulated. Then a special neural center in the medulla oblongata. Yes, it is also correct to regulate cardiac activities. Adrenal medullary hormone, sympathetic nervous system based neurotransmitters are produced from adrenal medulla. So like epinephrine, norepinephrine. So these three are useful for regulation of cardiac activities. So many will get confused with this D also, adrenal cortical hormones, cortisol, because in that last chapter they had given that cortisol is also having a role in regulation of heart functions, but that is about maintenance only, no need to apply here. So we had gone with option 4, A, B, C only. Okay, so for 173 question we have to opt option 4.
Uh, 174. Question number 174. Please see. Assertion. A typical unfertilized angiospermic embryo sac at maturity has 8 nucleated and 7 celled structure. That is absolutely correct. If we draw it like this, it will come something like this. This is the central cell and these are the antipodal cells. Okay, so this is correct. 7 celled, 8 nucleated. Now reason, the egg apparatus has 2 polar nuclei. This is wrong because polar nuclei are in the central cell. The 2 cells of egg apparatus are called as synergids, not polar nuclei. So assertion is correct, but reason is wrong. Fine. Okay, so let us see. Assertion is true, but reason is false. Answer 2. Fine. Next, 175. Find a statement that is not correct regard to the structure of monocot stem. You should know monocot stem, the vascular bundles are scattered, they are conjoined, collateral and closed. So vascular bundles are scattered, this is correct. Vascular bundles are conjoined and closed, this is okay, correct. Phloem parenchyma is generally absent in monocots, this is directly NCERT line. And hypodermis is parenchymatous. No, hypodermis in monocot stem is sclerenchymatous and hypodermis of dicot stem is collenchymatous. So hypodermis is not parenchymatous, but hypodermis is sclerenchymatous. So our answer is this, because this is not correct for the monocot stem. Fine. Next. 176. Given below 2 steps, both wind and water pollinated flowers are not very colorful and do not produce nectar. Yes, because wind and water pollinated flower, they do not have any biotic agency. So no attractants are required. Attractants are required for biotic agencies. So this statement is correct. Reason, the flowers produce enormous amount of pollen grain in wind and water pollination. So see, reason is also correct, but not the correct explanation. Why large amount of pollen grains is produced? Because it can be randomly distributed. So the chances of pollen grain falling on wrong places is more. That is why more pollen produced. So reason is correct. Assertion is correct, but reason is not the correct explanation of assertion. The correct explanation of assertion can be that since the pollinated by a wind and water does not use biotic agencies, then it would have been correct explanation. So you see, both A and R are true, but R is not the correct explanation of A. Fine. So this is 176. Okay, then 177, I think it is one.
Question number 177 is from Health and Diseases chapter, Human Health and Diseases. Concept is cancer. Neoplastic characteristics of cells refers to. So neoplasm means actually when normal cells lose control over the cellular growth and cell divisions, then the abnormal cell growth will be there, abnormal multiplication of cells will be there. That time we can see neoplasms, means tumors. Okay, so definitely that particular neoplasm means abnormal group of cells will be having a mass of proliferating cells, continuous multiplication of the cells, because they don't have control on cell divisions. The next one is saying that, so I consider This is the characteristic feature of neoplasm. Okay. So which is cancerous. Next one, rapid growth of cells. What to say? Uncontrolled growth, proliferation is continuous. So abnormal growth is this. So rapid growth of cells also we can consider here. And next, invasion and damage to the surrounding tissues. Definitely, because metastasis property is there, and also cancer cells will absorb all the nutrients and they don't allow the neighboring cells to be normal. Okay, they absorb all the extra nutrients and all that, so that surrounding tissues will be damaged. Okay, so fourth one is like those confined to original location. Like generally benign tumors are confined to original location, not the malignant tumors. So that's why A, B, C are correct, but not D. So A, B, C only. Okay, option 1 is correct for question number 177. Okay, children.
One 78, the complex second of mitochondrial electron transport chain. This complex one is, you know, it is called as NADH dehydrogenase complex. Complex second, where when succinate is converted into fumarate, FAD is produced. That FADH2 is oxidized at complex second, and that is produced due to succinate dehydrogenase. That is why complex second is called as the succinate dehydrogenase complex. So this is complex 2. Complex 3, if you remember, it is called the cytochrome BC1 complex, and complex 4 is called as terminal oxidase complex. So fine. So these are the 4 complexes for ATS. Fifth complex is for the ATP synthesis called as F0F1 complex. Now, polymerase chain reaction amplifies DNA folding. This is very simple, children. When every time the amount is becoming double, it means you have to use the formula 2 to the power n. As you use in normal DNA replication, suppose a DNA has replicated 3 rounds, how many DNA molecules will be formed? So you apply the 2 to the power, something like this. Okay, so same here, every time it will become double, so we will apply 2 to the power n here. Fine. Okay, next. Here you see. In above represented plasmid, an alien piece of DNA is inserted at echo R1 site. So they have inserted here, alien piece of DNA they have inserted here. Which of the following strategies will choose the select recombinant colonies? Now due to insertion of the gene in the beta galactosidase gene, there will be no production of blue color. As a result, the recombinant colonies will be colorless. So let us see here the white color colonies will be the selected. So these recombinant colonies will be white color colonies. Fine. Okay. I think that's all. So children, that's all. So I hope you have understood all the questions. Of course, question paper was a bit more standard than the previous year, but mostly it was NCERT based. And
Yeah. With this biology question paper discussion is over, and we will we will hand it over to chemistry teachers. Chemistry teachers will continue the paper discussion. Okay, children. Wish you best of luck for your best result. Okay. God bless you. Bye.
अपन ऐसा करेंगे रैंडमली चलते हैं रैंडम किसी से कोई कोई आप हटाओ हमें हटा ले इसीलिए हटा लिया चलो तो भाई चलो बायोलॉजी में 91 खाली बायोलॉजी का अलग से है क्या कभी मेरी केमिस्ट्री में तो Physics also not sent in that group. Mic off. Okay, let us start the paper discussion. Question number 46. Ferromagnetism is considered as the extreme form of paramagnetic. You knowing paramagnetic means which is going to attract the magnetic field, and ferromagnetic is going to attract magnetic field very strongly. Mean this is correct statement. The number of unpaired electrons in chromium 2 plus. What is given here? Chromium 2 plus. If it is chromium 2 plus means it is 3D 4 configuration. 4 unpaired electron will come. And next is given neodymium 3 plus. It is going to be F3. 3 unpaired will come. Means this is wrong statement. Okay, next. Question number 47.
This question 47, there is a reaction given to us. It says A giving you 2B, both are gaseous, and they say the backward reaction rate constant is less by a factor of 2500. It's not printed there. Nevertheless, it's like say it is less than the factor of 2500 at 1000 Kelvin. Now, see, we all know that for a given reaction, like say A giving you 2B, we know RF is equal to say RB, right, at equilibrium. And we also know what is RF? RF is like say KF into concentration of reactant equal to what? KB, rate constant KB, and into B square. That's called law of mass action. Now I can say that KF upon KB will be equal to B square upon A. This is called equilibrium constant. Now they say this thing is 1 by 2500, 1 upon 2500. This thing is nothing but our Kc. They're asking in the question what is the value of Kp. Now to find out the value of Kp, once we have got Kc, we know that Kp is equal to Kc into RT to the power of delta Ng, right? Delta Ng is the difference of the gaseous molecules of product and reactant. So Kc is 1 by 2500 and R is given as 8.314 into temperature is, sorry, my mistake. It will be like say 0.0831 into 1000, right? So when you will solve it, you will get 0.033. That's the answer of yours, right? Next one.
Question number 48. In question number 48, we have to find the total number of isomers. It is about C4H8O. It is C4H8O. How many possible isomers means here structural as well as stereo. Cyclic structures we have to draw. So firstly, we will write the cyclic structures for C4H8O. First one, this one with 5-membered ring. All carbons utilized. Next one with 4. Now with 4, one carbon we can write one CH3 here. Other possibility in 4 is this one. If you write any other further, it will be repeated. Different structures I am writing. Next possibility, 3-membered ring. 2 carbons used. One possibility, ethyl, C4. Next possibility, dimethyl. And another possibility is, it is 1,2, it is 1,1 dimethyl. All possible structures written. Now in each structure, we will check where stereoisomerism is possible. This one is only one structure. No stereo possible. Here, no chiral center developed. Here, it is also 1. In this case, it's a new chiral center. So we will mark it as 2. Dextro, levo, or R or S. Here, this one is also a new chiral center. How many here? 2. Here, this is can be cis possible and trans possible. Trans will be optically active. So when it is optically active, we will take it as 2, which are enantiomers possible, and cis will be optically inactive. So this isomer, if you check the notes, this is done in the notes with cyclopropane example, 1,2-dimethyl cyclopropane. So how many total? 3. And here no stereo possible. So when we do it, 1 plus 2, 4 plus 2, 6 plus 3, 9 plus 1, 10. The correct answer is 10. Stereo is included. Okay, next one.
Yes, Mr. Like next 3. Question number 49. A hypothetical diatomic molecule with bond order 0 is quite stable. You are knowing if bond order is 0, molecule is going to be unstable. So this is wrong statement. As bond order increases, bond length increases. This is also wrong. You're knowing if bond order is going to increase, bond length is going to decrease. So here, statement 1 and 2 both are wrong. Question number 50. Here it is given molecule is ester. You can observe ester functional group is given, and this is converted into the aldehyde functional group. For ester to aldehyde functional group, we are knowing standard procedure. We have to use a dibal H, diisobutyl aluminium hydride. So you can observe first option is written here. It is Al. IBU twice means diisobutyl aluminium hydride followed by the addition of water. So first option will come. Next one. It is given the major product of the following reaction. Very good question. Here you can check the writings. Excess of CH3 MgBr. Excess of CH3 MgBr. Okay, to solve this question, I will write the structure of this molecule in a proper way. This is given pH, C double bond O, C triple bond N. And now in the question, they are writing Grignard's reagent CH3 MgBr is taken in excess. So here it is a CH3 MgBr. It is in Grignard's reagent Mg is plus and CH3 is having delta minus charge. Since excess is given, I will perform reaction of Grignard reagent with this ketone functional group as well as a nitrile functional group. Here, this carbon is delta plus and oxygen delta minus. This carbon delta plus and here nitrogen is delta minus. So one I will attack this CH3 minus on this double bonded carbon and pi bond will break. O minus will bind here. O minus double up where MgBr is going to bind. Second, I will take one more molecule of this to attack on this carbon also and pi bond will break. It become N minus. By this reaction, I will get, for this I am representing by pH. This becomes CO minus MgBr and here CH3 of Grignard's reagent bonded with this carbon. Then this nitrile is given, 1, 2, this nitrile is given, and for this nitrile, I will bind one CH3 and it is double bond N MgBr. When I'm doing hydrolysis, this is the second part of the reaction, this bond will break and here oxygen will bind, and this bond, this will get hydrolyzed or acidified, and this O minus will take H plus from acidic water, it become OH. So this is OH and one CH3 group is coming here and it is ketone with one CH3 group. So you can check options now. Excess Grignard's reagent was given. So yeah, correct answer will be, correct answer is, first one only the correct answer. Okay. Here this part become alcohol and this part become ketone. Next, question number 52.
Well, in this question, there is a correction. A data is missing. Let's say that acid is basically HA. HA, they have given the data of the molar conductivity of the A negative is given as 50.4, right? And the molar conductivity of H ions, which are missing at infinite dilution, the data is 349.6. And we all know that molar conductivity at infinite dilution of this acid HA will be equal to sum of the ions, right? It will be 400. Now we all know that alpha is equal to molar conductivity at given concentration upon molar conductivity at infinite dilution. When you put the values are, this is 90, this value is given as 90. So I'll make it 90 upon 400. When you will solve it, you will get 0.225. Hence the answer 0.225, right? So the next question now we have.
In next question, it is from the question number 53. It is from the qualitative analysis, organic chemistry. Which of the following reactions does not belong to Lessing's test? This is direct reaction base. Sulfur, it is converted sulfide, direct reaction of the Lessing test. Identification of the ion. Halogens directly converted to halide ions. And nitrogen with a carbon converted to cyanide. Among them, which is not part of the Lessing test is third reaction, CuO. Okay, third one. Then 54. 54 it is from the carboxylic acid chapter, that acidic strength concept. In this case, as ERG increases, the acidic nature also increases. So among them, among the aliphatic acids, the most strongest will be which one? Formic acid without any alkyl group. So here if we go by the options, first and fourth option eliminated. Between 2 and 3, we go by that. As ERG increases, acidic nature decreases. So here among these two, after formic, here 1 methyl, here how many? Here how many are there? 3 methyl groups. More ERG where between 2 and 3 here, lesser will be the acidic nature. So correct answer should be which one? Second option. Right? Now, sorry, sir.
Oh, Tawfiq sir, chala shock coming. So yeah. The next is from biomolecule. It's an easy question if you check the options properly. Because this table people don't remember much from class 8 onwards, they are trying. But if you see, people remember D, rickets, which is there for everyone. B third, in all options B third is there. You can match vitamin B6 with conversions, B2 with chelosis, but at least B6 with conversion, you will get the answer directly. D second, this option is coming. And then C first, so clearly answer is the first option. A fourth, B third to every option is there, C first and D second. Next, a little bit kind of controversial question because of probably the printing mistake as done regularly by Neet examination. Out of the following complexes, which are the following compound will have the minimum conductance in the solution. So we say that conductance is proportional to the number of ions we say and charge of the ions we say. So at this point of time, we have to see only the number of ions. First, we'll get the answer directly. So there are 3 + 1, 4 ions here, 1 + 1, 2 ions here. There is no ion here. And this is also not having any ion, probably because there should be one ion outside, but they forgot to mention it. So right now, 1 and 4 both appear to be having almost zero conductivity, not zero, we should say, little bit conductivity every solution will have. H+ or H- ion will be there in aqueous solution every time. So 1 and 4 will have the minimum conductance, we can say. But out of that, the better option would be the fourth one, assuming that NTA would have done a printing mistake here in the first option. So the best option here at this point of time, little controversial, but will be the fourth option. Next, 50. Clean, keep. पहले clean करना पड़ेगा. So this was also controversial. Not the question, but the change of question. Next, sugar X is found in honey. So if you see just in honey, so glucose might also come in the head. So next, keto sugar. When keto sugar comes, so glucose cannot be the option. Generally, by B, keto sugar, people will get the answer should be D, fructose. Alpha, beta anomeric forms are there. Levorotatory, fructose. Then correctly the answer will come D, fructose. For next questions, Tawfiq sir.
Question number 58. How many products including stereoisomers are expected from the monochlorination reaction of the following compound? In this compound, we have how many types of hydrogen? There are 4 types of hydrogen. So we will get how many monochlorostructural isomers? 4 monochlorostructural isomers will be there. One possibility. Second possibility, third possibility, and fourth possibility. These are 4 structural isomers. Now, stereo means in each structure you will check for the where stereoisomerism is possible, which is here optical. In the first structure, this is not a new chiral center, that's why it is only one structure. In second one, there is no chiral center. Sorry, here it is. New chiral center, it is CH2Cl. This is new chiral center, so this will be new chiral center counted as 2 dextro or levo. Here, not a new chiral center, it is only 1. Here it is a new chiral center, it is how many again? 2 dextro levo. And here it is normally no new chiral center, it is 1. So here 2 plus 1 3 plus 2 5 plus 1 6. Total 6 isomers are there, which means including stereoisomers. So correct answer is option number 3. Next one is, which of the following compounds can exist as cis-trans isomers? This is from the conditions of cis-trans isomerism. We apply that one, that here, structure 1. 2-methyl hex Now, in that cis-trans or geometrical isomerism, restricted rotation is there, but at each end, there must be two different groups here. There are two same groups. It will not participate in GI. Next one, 1,1-dimethyl cyclopropane. In cyclopropane, 1,1-dimethyl. Here, restricted rotation is there, but here also two same group, here also two hydrogen, here also two hydrogen, not satisfying the condition. So this option also eliminated. Third one, 1,2-dimethyl cyclohexane. 1,2-dimethyl cyclohexane. In this case, restricted rotation, yes. Now at this carbon, there are two different groups, one H and one methyl. On adjacent, one H and one methyl. So here we will get cis isomer, both CH3 or both H same side. Other will be trans isomer, one CH3 above, one CH3 below. It is satisfying the condition of GI. Third will be the correct answer. Similarly, pent-1-ene, it has restricted rotation, but at the end there is CH2. It will not go for geometrical isomerism. 60th question, which of the following reactions does not give benzene as the product? First reaction, N-hexane, and this is which reaction from alkanes? Aromatization reaction. After this, overall, it gives which product finally? It is giving the benzene formation. So question is, which will not give benzene? Option 1 eliminated, aromatization. Second one, again from hydrocarbon chapter reaction, this is cyclic polymerization of alkyne. This results into again benzene. First and second options eliminated. Then third one. Third one from which chapter? Phenols. Diazonium salt, H2O, warm water or dilute acidic medium. It results into which one? Finally, phenol formation. And last one, sodium benzoate, directly in benzene chapter given hydrocarbons, soda lime. Answer will be benzene. So among them, the best answer is, or clear answer is which one? Third one. Okay.
Well, the next question. Right. Up. Change. Right. So in this question, I mean, they have given a phosphoric acid and they have also given the ionization constants of them, right? So if we write down the three equations, H3PO4 giving you H plus and H2PO4 negative, and again H2PO4 negative, giving you one more H ion, and HPO4 2 negative, HPO4 2 negative, dissociating to give you H ion, and PO4 3 negative, right? Now we all know that it is difficult to take out the H ions from a negative charge, right? Ion which is having a negative charge, right? As we go down, the amount of negative charge keeps on increasing. So it becomes even more difficult to take out the H ion. So definitely the dissociation constant or ionization constant, as we keep on going down, it will keep on decreasing as well. So I believe that this statement B is correct. Now, if we'll add them all these things will cancel out and we'll get H3PO4 giving you 3 H ions and PO4 3 negative. But once we add the three equations, we all know that the equilibrium constant, dissociation constants, all can be multiplied. So K1 into K2 into K3, right? Now, if I take logarithm of that, log of K will be equal to what? Log of K1 plus log of K2 plus log of K3. We are utilizing the property of logarithm when we have log of ABC is equal to log of A plus log of B plus log of C. So this one is as well correct. Now we all know that as we keep on going down, the dissociation constants keeps on decreasing and they are decreasing. So from my side, A, B, C, which will be A, B, C, third option will be the right option, right? Now let's come to the next question, which is 62, right? They're saying find out the molecules out here which contains same number of atoms, right? So let's do that. Let's take the first option. They have given the weight of that, Na2CO3. So number of atoms in Na2CO3 will be first of all what? Number of moles into Avogadro number into atomicity, right? So number of moles will be 212, the weight given upon the molecular weight 106 into Na. And what is the atomicity of this molecule? 6. So into 6, this happens to be 2. This is 6 into 2, 12. 12 Na. Right. Let's come to the second option. 248 gram of Na2O. So number of atoms out here will be again the same. Number of moles into Avogadro number and into atomicity. So number of moles will be weight given 248 upon the molecular weight happens to be 62 into Na and the atomicity. What is that? 3, right? So 62 into 4. 4 3s are 12. So 12 Na again out here. Okay. Let's try the third option now. So again, the number of atoms will be weight 240 upon molecular weight 40 into Na, Avogadro number into atomicity, which happens to be 3. So here this will be 6. 6 into 3, 18. 18 Na. Now definitely 18 Na is not equal to 12 Na and 12 Na out there. So let's check one more option called D. Number of atoms here will be again weight upon molecular weight, that is 2 into Na, and the atomicity, which happens to be 2. So this is 6, 6 to the 12, and again 12 Na. And if we look at the options right now, where the options, well, okay, I'll solve the last one as well. 220 gram. Number of atoms here will be weight upon molecular weight of CO2, 44 into Na into atomicity, that is called 3. 5, 5 3s are 15. So this is 15 Na. I believe only first one, second one, and this one, which is called D. A, B, and D is the right option. A, B, D. The first will be the right answer, right? Next one, sir will take.
Oh, current mark de do, sir.
Current also.
So shocking paper it is. Everyone is giving shock. So one small gap I will take here to tell the people, those who are getting demoralized, don't worry. You'll be surprised with the cutoff. The least you can imagine, lesser than that the cutoff is going to go, don't worry. Let's complete the paper discussion part first. So yeah. Like nitrogen, that can form ammonia, arsenic can form arsine, correct? NH3, PH3, SH3, SbS3, BiH3, you have done already, all the properties you have studied. Yes, it exists. Antimony cannot form antimony pentoxide. Very specifically about antimony, they are asking to create the doubt in the mind of the student, but as per NCERT, all the elements in nitrogen family form 2 types of oxide, E2O3, E2O5. So keeping that in mind, antimony can form antimony pentoxide, so second should be false. So first is correct, second is false. So first correct, second incorrect. Second option will go. Dalton's atomic theory that could not explain which are the following. Reading of NCERT in physical chemistry is taken as something which should not be done outside Goshalite. But here we again and again tell you, read NCERT, questions will come from there. It's not only numerical sciences. Physical chemistry has a lot of concept on which the questions can come. Like the Dalton atomic theory. This exact line you will find in the NCERT. Dalton atomic theory, after 4-5 points of NCERT of Dalton atomic theory, which they are explaining, atoms are indivisible and all. After that, the paragraph is there that it can explain the law of constant proportion, multiple proportion, and conservation of mass. It cannot explain the law of gaseous volume. Why and what? That is not the point now. I don't think you will need the answer of that as well. You just need the answer of this question. Answer is 3. Well within the boundary of NCERT, some basic concepts of chemistry NCERT you can open. Some basic concepts of chemistry, Dalton atomic theory. Very good photograph of Dalton uncle is there. After that, this theory is mentioned over there. Then 65, sir will discuss. 68, meanwhile, one question is there. I can wind it up. The standard heat of formation in kilocalorie per mole of barium 2+ is, they are asking. Standard heat of formation of sulfate ion they have given you. So standard heat of formation of sulfate ion is given minus 2.6 kilocalorie per mole. So enthalpy of formation of sulfate ion I am writing. That is minus 216 kilocalorie per mole. Standard heat of crystallization of barium sulfate solid. This can confuse a few people if you have not seen the definition properly. Standard heat of crystallization they are saying is minus 4.5 kilocalorie per mole. We'll come to that. Standard heat of formation of barium sulfate. They are giving you as enthalpy of formation of barium sulfate. So enthalpy of formation of barium sulfate is minus 349 kilocalorie per mole. Everything is in kilocalorie, so no problem of units. They are asking me, giving me enthalpy of formations, these two, and they are asking me enthalpy of formation of barium 2+. So very clear cut, I am going to use enthalpy of formation of P minus R, product minus reactant. All I need is a reaction, and the reaction should be crystallization of barium sulfate. Now, barium sulfate solid, if I take and put that in aqueous medium, water, I will get barium 2+ ion aqueous plus sulfate ion aqueous. This is called enthalpy of solution of barium sulfate. Reverse reaction is crystallization. Sir, where we have done this? 100% you have done here. Ionic, you have done the same thing. Forward reaction is called solubilization. Backward is called crystallization. So, bhaiya, this is the enthalpy of crystallization they have given as minus 4.5. So, enthalpy of solution will be 4.5. So, 4.5 is equals to enthalpy of formation of product minus reactant. Answer will come. Product is what? Barium ion, which is asked, sulfate ion, which is given, and barium sulfate, which is also given, minus 349. Put the value, I think around 128.5, something like that, the answer will be coming. So, direct question is there. Only thing was heat of crystallization, how to understand. That's why the paper is good. We cannot say it's a easy paper from any angle. Moderate also we should not say. Next, VSP sir, can you please take? Shock, one more.
Question number 65, directly from NCERT. The correct order of decreasing basic strength of the given amines. First is given N-ethyl ethanamine, means it is C2H5 twice NH. Ethanamine means C2H5NH2. Benzenamine PHNH2 and N-methyl aniline PHNHCH3. In case of basic strength, you are knowing these first two are aliphatic amines. They are more basic than aromatic. Out of first and second, this I am writing 1, 2, 3, 4 like that, so I can easily explain. Out of 1 and 2, you're knowing directly. You're knowing fact also. In case of ethylamines, it is 2, 3, 1. So 2 degree, 3 degree, 1 degree. So first is more basic than second. In 3 and 4, if we check, this CH3 is plus I group, which is going to increase the basic strength. So 4 is going to be more than 3. Okay, and why 3, 4 are less than 1 and 2? Because in case of 3 and 4, lone pair is delocalized. While in case of 1 and 2, it is localized. So obviously localized is going to be more basic than delocalized. So based on this, you can check this third one is benzenamine is minimum. So only one option available. Next question. 66 discussed or not? Okay. Unlike gallium has very high melting point. You're knowing gallium has low melting point, is roughly 30 degrees Celsius. That's why it is used in the high temperature measuring thermometers also, because melting point low and boiling point high. So they're saying unlike gallium that has very high melting point. This statement is wrong. Gallium and cesium both have low melting point, roughly 30 degrees Celsius. So this is wrong. Second, on Pauling scale, electronegativity values of nitrogen and chlorine are not the same. This option is not having any significance in this question, because this is not going to decide answer here. So I will leave this thing. Argon, potassium plus Cl minus calcium 2 plus sulfur 2 minus are isoelectronic. You're knowing how to count electron and everything. If electron count equal, obviously they are isoelectronic. This is correct. The correct order of first ionization enthalpy of sodium, magnesium, aluminium, silicon. So if you have sodium, this question many times came in our papers. Sodium, magnesium, silicon, sodium, magnesium, aluminium, and silicon. Magnesium is going to be more than aluminium, because this is having 3s2 configuration and aluminium is 3p1. So actually it is sodium, then aluminium, then magnesium, and then silicon. So they are saying sodium, then magnesium. So this is wrong. Atomic radius of cesium is greater than the lithium and rubidium. This is well-known fact. So C and E are correct. Question number 67. You have to match them. XCO3. You are knowing in case of XCO3, it is sp3 hybridized trigonal pyramidal structure. So sp3 hybridized trigonal pyramidal or pyramidal written here. XCF2. XCF2 is linear molecule having 3 lone pair at the central atom, and this is linear hybridization sp3d. So sp3d linear. XCOF4, this is the structure, okay. sp3d2 and square pyramidal. And XCF6, it is discussed many times, it is distorted octahedral, also named as capped octahedral hybridization sp3d3, okay. So now you can match answers there. Question number 69. Among the following 1, 2, 3, correct order of bond dissociation energy of CH bond marked with the star there. This one, all cases it is CH bond. In first one, it is sp2 carbon. This case it is sp carbon, and this case it is sp3 carbon. All cases it is carbon hydrogen bond, but hybridizations of carbon are not same there. And you're knowing as we are moving from sp2 to as sp3 to sp2 and then sp, percentage S character is increasing. And because in the increase of percentage S character, bond length is going to decrease and bond energy is going to increase. So in this case, this is sp carbon hydrogen bond, and they're asking about the bond dissociation energy. So sp carbon hydrogen bond is going to have maximum bond energy, followed by sp2 carbon hydrogen bond and then sp3 carbon hydrogen bond. So answer is a 2, 1, 3. Next one, question number 70. Predict the major product of the following reaction. Very good question. This compound is given, and first reaction is given there HBr in the presence of benzoyl peroxide. HBr in the presence of peroxide. This is going to produce bromine radical, which will cause homolytic bond fission. And you're knowing radical is more stable on this 3-degree carbon, and that can be generated if bromine bind to this carbon. So this first step of the reaction or first part of the reaction is giving this product. Then second is given there KCN. If KCN is given, this bromine is good leaving group, and when KCN is given, this bromine is going to leave the substrate and will get this compound. And next one is given there sodium amalgam in the presence of ethanol. Sodium amalgam in the presence of ethanol is good reducing agent, and this will reduce the CN into CH2NH2. So this CN nitrile is reduced into this CH2NH2. So you can find answer there. Okay. So which option? Let's check it. So answer is third one. Next. अच्छा 4 है नीचे लिखा हुआ. Okay, sorry. 4 is written here down. Okay, this option, this one. Next one. Haber process, you're knowing Fe catalyst is used. Wacker process, PDCl2. Wilkinson's catalyst, RHPPH3Cl, and Ziegler-Natta, you're knowing TiCl4. Okay, so you can easily match, is directly match there. Now LC sir will continue here.
Thank you, sir. Well, next one is like say bit calculative, but not so. See, they're asking in this question the radius of, they're asking the radius of an energy of the first Bohr orbit of helium and lithium 2+, right? So we know the formula for the energy En is equal to, from this perspective, it is minus of 2.18 into 10 to power of minus of 18 into z square upon n square, right? Since they're asking for the first orbit, n will be basically 1. Now, if I want to find it out for, say, lithium 2+, I'll put the value of z of lithium, that is 3, so 3 square. So minus of 2.18 into 10 to power minus of 18 into 3 square, 9. When you will solve it, you will get it like, say, I believe you'll get 19.62, minus of 19.62. And if you want to find it out for helium plus, it will be same, minus of 2.18 into 10 to the power minus of 18 into Z square, that is just 4, right? So multiply 4 into this one and you'll get 8.72, minus of 8.72. Now there is no need to go for radius because there is no other option matching with that stuff. So correct answer will be fourth one, right? So make sure that you are having the powers corrected over there. Okay. Next one, Tawfiq sir.
Question number 73, assertion and reason question. First one. This compound, which is a primary alkyl halide, which halogen given iodine, undergoes SN2 reaction faster than another compound, which is also primary with Cl. So we know that among these two, which is faster in reaction, first one. So first is correct. Why? Because the leaving group ability of iodine is better than chlorine. Reason given, iodine is a better leaving group because of its larger size. So we are deciding here that I minus is a better leaving group, and we know generally the weak bases are better leaving groups. So out of I minus and Cl minus, which is a weaker base, I minus. So among these two, among that reason, it is correct that I minus is a better leaving group, and that is how we have decided the assertion. So here answer will be which one? Option number fourth. Assertion and reason are true, and R is the correct explanation of A. Then, yes, Girish.
So the next question is 74. If they're asking, 99% time taken, to complete 99.9% reaction in terms of half-life. We all know that T 99.9% is equal to 10 into T half. T half is given 1 minute, or 10 into 1 will be 10 minutes will be the right answer out here. And out here, which is the following aqueous solution will exhibit highest boiling point. Now delta TB is proportional to I into M, right? So more is the value of I into M, more is the value of delta TB. So let's say the value of I into M out here. So I will be 2.01, that is 0.02 out here. I will be out here 3 and into 0.01, that is 0.03. And I over here for this glucose will be 1 into 0.015 equal to 0.015. And for urea, again, it is a non-electrolyte. The value of I will be again 1 into 0.01, right? So I believe this 0.03 is the highest value. So the answer will be Na2SO4, second one. Now coming to 75. 76. Right, 76 is a question from chemical equilibrium. Now look up the value. This value is plus. That means this reaction is endothermic reaction, and for endothermic reaction, the reaction will proceed forward if we increase temperature. So first option is definitely correct. Now out here, if I increase the amount of reactant, reaction will keep on moving forward. So if I want the more amount of NO, I will have to put in more amount of N2 as well as O2. So C and D both will be the answer. So from my side, this is A, C and D. A, C and D only, right? Next one.
Very easy question, not taking much time of yours. That's a question of practical chemistry part. They are asking which group it belongs to. So you can directly get the answer by matching just Al3+. All Fe-Car, Al3+, Fe3+, Cr3+, they are group 3 cations. So if they are group 3 cations, and D will match with third group, that is D, second option will match. So first option is coming as the answer. I think group theory the people might be knowing, not going to be much of a problem. 78.
Now this 78 is they're asking about the wavelength. And we all know
Mark,
right? We all know that 1 upon lambda is equal to RZ square into 1 upon N1 square minus 1 upon N2 square. Now when we take the ratio, they're asking ratio. So this part will cancel, this will be common for hydrogen. Now we have to find out this value. So when we'll find out 1 upon lambda 1 for this transition from N2 to 3, right? So this will be 1 by 4 minus 1 by 9 happens to be like say 5 upon 36. So I believe lambda 1 will be 36 by 5. Similarly, if we find out 1 upon lambda 2 for this transition from 4 to 6, it will be proportional to what? 1 upon 16 minus 1 upon 36, right? When you'll solve it, you'll get 1 upon lambda 2 happens to be like say 5 upon 144, so which means lambda 2 will be 144 upon 5. When we'll take the ratio of that stuff, lambda 1 upon lambda 2 will get 36 by 5 upon 144 by 5. So 5 cancels up. This is 1 by 4. So answer will be 1 by 4. Third. Yes, please.
Next question is the correct order of wavelength of light absorbed by the following complexes. This is question directly from the NCERT table. You can observe here C is given CuH2O4 2+. It is very interesting example because this compound is blue in color but is going to absorb red. And red is the maximum wavelength in visible region, so this must be maximum here. So option number 1 or 4, C is maximum in these two cases. These two can be eliminated. Now, in this case, you can check ammonia, CN and H2O given. Here, CN is a strong field ligand. And since a strong field ligand, it will create maximum CFSE. And if crystal field splitting energy is high, lambda absorb is going to be low. So B is going to be least. Again 1 and 4 tie there. Now we have to check A versus D. In case of A, you can check ammonia ligand is taken, which is stronger than H2O. If ligand field is stronger, CFSE more, lambda less, so A is less than D. So here D more and A less, so correct answer is first. Next, identify the correct order against the property given here. H2O, ammonia, CH3L3 dipole moment order. You're knowing dipole moment of water is more than ammonia and ammonia is more than chloroform. This is well-known fact discussed many times in the class. So this is correct. Second is given Xc. The question is number of the lone pairs on central atom. In case of XCF4, we have 2 lone pairs on central atom. XCO3 1 and XCF3 it is 3 lone pairs. So this is not matching. It's wrong. OH, CH and NO given, you can observe here, OH, it is oxygen second period, hydrogen first, second first, and these both are second period. This is going to be maximum bond length, and they are saying minimum, so this is also wrong. So A is correct, B, C, wrong, and next one is bond enthalpy. N2 means nitrogen, nitrogen triple bond, O2 means oxygen, oxygen double bond, and H2 means hydrogen, hydrogen single bond. So NN triple bond energy is more than double bond and double bond more than single bond. It is very obvious. If it is triple bond, more energy required to break. So A was correct and D is also correct. So you can go for fourth option here. Next is given. They are asking question from the methods of purification of the organic compounds. If chloroform aniline mixture is given, it can be separated by the simple distillation. So for A, it is a fourth one. Crude oil in petroleum industry, it is separated by the fractional distillation. Glycerol from spent lye, distillation under reduced pressure, and aniline water mixture, steam distillation. Okay, so for A it is a fourth, 1 or 4 option. For B 3, so ignore. C, for C it is 1, so C fourth option. Okay. Okay, I'll complete this 83 also. Which of the following electronic configuration belongs to main group? Main group means S and P block elements. So first is given 3s1 S block element. This is correct. It is given 3d3 S2 means D block. So this is not main group. D10 S2 P5. P5 given here. It is P block element. So this is also main group. D10 4s1. It is D block, not main group, and this is also not main group, it is a D block. So it is A and C which are main group element or S and P block element. Next. Okay, so question number 82.
So question number 82, it's a first order reaction, and for first order reaction, time taken is equal to 2.303 upon K log of A naught, initial amount upon 80, right? Remaining amount. Put the values out there, 2.303, K is given as 0.03, log of A naught is 7.2 upon 0.9, right? Solve it, this is log of 8. Log of 8 is how much? 0.9. So 2.3 into 0.9 upon 0.03. Solve it, you'll get 69.3 seconds, right? 83, 84. 84 is comparatively simple one. We have got humidity and we are looking for type of solution. What is humidity? Water vapors in the gas. So we will have liquid and gas out here. Alloys are what? Solid and solid. And I believe that will be like say comparatively okay with us. A will be like say second. A second. We have A second here also. B will be 1. B will be 1. So first is the correct answer. No need to further check it up. Sorry. 85th is solution chapter. Now we see that they're asking that this solution is like having a positive deviation, negative deviation, or like say it's an ideal solution. So let's say we are having 5 moles of liquid X and 10 moles of liquid Y. So if you look at the mole fraction of liquid X, mole fraction of liquid X will be like say 5 upon 15, that is 1 by 3. So mole fraction of liquid Y will be basically 10 upon 15, happens to be like say 2 by 3. Now let me find out the value of PA plus PB. So what is value of PA plus PB? This is P naught A into xi A plus P naught B into xi B. I'm assuming this is A and this is what B. So values will be like P naught A is given as 63. 63 into 1 by 3 plus 78 into 2 by 3. Solve it, you will get almost 73. Now this 73, this PA plus PB, which is 73, is more than basically that the PT one, right? So PT is 70. That means PT is less than PA plus PB. This is the case of negative deviation. So the answer will be, the solution shows negative deviation. First one, right? Now out here in the next question, this is simple one. They're asking that which of the following represents delta H minus of 74.8. Now this is the difference between the energy of reactant and product. But if you look at these two options out here, these are the energy of activation. Energy of activation, so clearly they will not be the answer. Now if we look at the third and the fourth option, basically like this is endothermic, they have given minus of that, so we need an exothermic reaction. I believe that this will be the right answer. Okay, तो fix.
जाते जाते Question number 87. Which of the following compounds does not decolorize bromine water? Bromine water test, in the test of functional groups we have done that one, or in the phenol chapter first one. Phenol with bromine water, it will decolorize and becomes tribromophenol. Which option eliminated? First one. Second one, it is a hydrocarbon chapter reaction. The double bond will be, addition takes place, electrophilic addition, and it will decolorize the bromine water solution. First and second, both eliminated. Third, aniline. Aniline reacts with bromine water, giving tribromoaniline, white precipitate, just like first option. So first, second, third, they all will decolorize. Which option will not decolorize? Simple, a saturated hydrocarbon, no multiple bond is there, and no such case of strong activating groups. Answer will be fourth. Then question number 89, which is there from the diazonium salt, amines chapter last part. In that these are diags.
2, 1, light. 21. 3, 87, you are live.
ठीक है. Question number 87, we have discussed it. Answer is fourth. We have no decolorization of bromine water. 88th, we have to find the oxidation state of the underlying elements. Potassium, alkali metal, always in compound state +1. Peroxide, oxygen is -1 each. and sulfur in H2SO4 is +6. Answer is which option? Option number 4. Then it is from the diazonium salt chapter, amines chapter, diazonium salt, last part. Diazonium salts are prepared by this station. Yes, temperature condition given correct, 0 to 5 degrees Celsius or 273 to 278 Kelvin, which decomposes easily in dry state. Exactly correct statement as per the NCERT lines given. Insertion of iodine in the benzene ring is difficult, and hence iodobenzene is prepared through the reaction of diazonium salt with Ki. This line given at the end of the amines chapter, that fluorine and iodine cannot be introduced easily on the benzene ring, so that line is correct. Then what is the easy way? Firstly, convert the benzene up to the aniline, reduction, diazonium salt, and then iodobenzene. Fluorine and iodine, both lines given there at the end of the amines chapter. Okay, next will be, please.
Okay, next is question number 90. Question number 90. Very, very, very good question. Just to check, you're just mugging up the facts or you're knowing concepts. Because many people are just mugging up the points written in the NCERT. They're not trying to understand the concepts there. And thus are doing mistake. Learn the concept, paper will be easy for you. Which of the following are paramagnetic? First is given nickel 2+ complex where Cl is taken, which is a weak field ligand. Ni2+ means it is a 3D8 configuration. 3D8 configuration. Since a weak field ligand taken, this will go for sp3 hybridization. And if it is sp3 hybridization, then this 3D8 configuration will not be changed. So I can write electrons like this: 1, 2, 3, 4, 5, 6, 7, and 8. Here it is having unpaired electrons, means this is a paramagnetic. Second, in NCERT they are given NiCO4 is diamagnetic. But if you are knowing fact, then you can solve this question properly. In case of NiCO4, it is nickel 0. It is a 3D8 4s2 4p0 4d0 configuration. Here electrons are present in 4s or ns. So we have to move ns electron in n-1d. If I move electron of 4s into 3d, it is going to be 3d10 configuration. And if it is 3d10 configuration, it is not having any unpaired electron, whether it is SFL or WFL. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. No unpaired electron means it is going to be diamagnetic. Next is given Ni2+ Cn4 2-. Here it is nickel 2+. Same Ni2+ D8, but here given ligand is a strong field ligand. Since it is strong field ligand, it will create pairing, and because of the pairing, it is going to be dsp2 hybrid complex. So out of 5 d orbitals, if I use 1 d orbital for the hybridization, dsp2 like this, okay, dsp2 like this, then I have only 4 d orbitals to fill 8 electrons. So 1, 2, 3, 4, 5, 6, 7, and 8. No unpaired electron means this is also diamagnetic. Next is NiH2O6 2+. It is a weak field complex and D8. So I can write like this: 1, 2, 3, 4, 5, 6, 7, 8. Here also unpaired electrons present. So it is a paramagnetic. And fourth one, NiPPS34. You can check. It is given NiPPS34. There is no charge on complex. PPS3 is also uncharged. Means in this case it is a nickel 0. And if it is a nickel 0, it is a 3D8 4s2 configuration. And if I move 4s electron into 3D, this is going to be 3D10. And if it is a 3D10, there is no unpaired electron. You can check it here. So this is going to be diamagnetic. So here I can say A and D are paramagnetic. So which option available there? Second option. Okay, chalo, now physics will be continued.
Yeah, easiest paper.
Hello, hi. It's too late, but we will finish out. And paper paper was not one of the toughest paper in the history of Neet or AIPMT, whatever you say. But once you can see the paper discussion, entire Gosalites team of physics will do the paper discussion. So we'll start with Mohit sir. For cutoff and everything, tomorrow we will have a separate meeting. There we can discuss what will be cut off, what is good marks in physics, what is good marks in chemistry and bio. Right now, we'll finish out fast and we will see physics 95 is a very good mark. So tough. Yeah, we are also feeling. We know our students, you know us, we know us. So of course this was the toughest paper, but parvaledu, it's a competitive exam. Relative marks is important, not individual marks. So don't worry. Let's start with the paper discussion. Our cool sir will start with paper discussion.
Okay, so we got here first question from your error, and probably all people have done this question. In this one, you got P equal to A cube B square by C, and this root D is given. And you know what to do in this one? We can write here 3 delta A by A, okay, into 100. Plus we can write 2 delta B by B into 100. Plus we can write here delta C by C into 100. And plus half of D, okay, del D by into 100. Now these values are given. And 3, this is your 1, plus 2, here we got 3, plus in this one del C, that is your error in C given 2 and half, and delta D by D, that is the 4. Now you know that 3, 6, 9 to 11, and this finally here you got 13%. So all have done this question at least correct. This was the, yes. Now, next question is here, question number 2 from your wave optics polarization.
So many questions like that came in physics exam.
Change. Okay. And now we have asked this question multiple times, this pattern, only one change is there. This angle, just to confuse you, given here 22.5, that is this practice you have done. So in this one, here is the one key point that the Hawaiian I naught is the intensity of the polarized light after passing through the first polaroid. We can write here, this is your first polaroid, and then this polaroid, they are crossed. And we are keeping one polaroid seat, and this one we are keeping at 22.5. Right now I'm saying this one is the theta here. Now we can say polar intensity after first is given here I naught. There are the chances of mistake here in this one. They are given in the bracket. Now you know if this is the I naught, here intensity becomes your I naught cos square theta. And now here this angle, if you find, this is going to be your 90 minus theta. So we can it finally it becomes I naught cos square theta into your cos square 90 minus theta. Cos square 90 minus theta will become your sin square theta. Now we can write, we need to make, because this theta is 22 and a half. And you know that this will be converted to 45. So what to do here in this one? You can write I naught by 4, and here you can multiply this 4. You can write like sin square theta into cos square theta, and now it will be visible to you. And this thing will be coming to your mind also, that 22 point you cannot solve. It means it how to convert to your 45. So here you can see this one, we can write as I naught by 4, and this one sin 2 theta whole square. And now you can write sin 2 theta. Theta here 22.5, that becomes your 45, and sin 45 1 by root 2 square of that one. So you can write here I naught by 4. This is the sin 45 square, and finally we can write becomes I naught by 8 in this one. Okay, so slightly tricky because angle was 22 and a half. Okay, but it gives feeling also that yet how to convert to 45 here. Now, yes. AKD sir.
So, oasis in the desert. One question came, easy question, but tough to find in the examination hall. Question number 3, they are asking you ratio of magnetic moment. Magnetic moment for current carrying coil, you already know, NIA, N-I-A. Or you can say number of turns, nothing mentioned. We can take 1 turn into I is same. area will be pi r square. So when you calculate the ratio m1 by m2, I will be same, pi will be same, so it will become r1 by r2 whole square. From there 1 by 2 whole square, that is very tough, 1:4. This is the question that everyone can do, and some of the question which everyone can do is this question. Let's move further.
So now you got this out of these three questions, or these three you can easily do. Next question from your unit dimension measurement again. And here in this one, they are asking, if you see here, that they are asking after zero error correction. It means some zero error is there. But suppose you didn't, if you didn't remember the zero error, then here, without that also using the common sense, you can see here that they have given in this one that vernier scale is at x equal to 0.1. It means directly you can subtract the reading if you are using the common sense. Otherwise, we can use here your main scale reading plus LC into your vernier scale reading minus of zero error. And here finding the least count is quite easy. So many times you have done at least this data. Each time you are doing in this one, 10 VSD is you're given here 9 MSD. You can write in this one 1 VSD equivalent to 9 by 10 of MSD, and then least count you will write here MSD minus VSD. So from here we are getting this will become MSD by 10. Main scale they have given 0.1, so we can write it becomes 0.1 by 10, which is the 0.01. Now this data you have to just put here. Main scale reading that is given 5 here. Least count just now you got 0.01 into your vernier. They have given this one coinciding vernier division is 8. So that is your vernier 8. And now in this one, zero error, okay, is 0.1. So you can write here in this one, this is the 4.9 plus 0.08. That is your second answer here. So yes, it is the question of zero error, but even though you didn't remember directly also, you know that with the common sense, you can subtract that reading, okay? So out of these 4 will not mark as these 4 to be very, very tough. Moderate question. Now, next we are having here select. Question number 5. Multiple time this was asked in your examination. All that time it was told that you have to remember relation for the means this de Broglie wavelength for any of the particle like electron, proton, and for the photon, you know. They're asking lambda for the photon. Always we use this formula energy equal to hc by lambda. So we can write here lambda photon is going to be your hc by E. And they are asking here for any of the particle. We always write that lambda is going to be your h by root of 2me. Now let's divide here. Lambda of photon divided by lambda of electron they are asking. And we can write here hc by E. And this becomes h, and here it goes 2me. H is getting cancelled. So finally you got here c root of, this is the root e, this is the e here, 2m by e, and this was the known question to you. And recently we asked in our one of the examination this one. So this question will mark as easy one here. Now we'll go for the next question, that is the from de Broglie. Common question in physics as well as chemistry. They are saying here in this one, de Broglie wavelength of an electron you have to find in the n equal to 2. So you know that electron only revolves, everyone knows this statement, 2 pi r equal to n lambda. So lambda you need to find here, you can write 2 pi r divided by your n. And the radius in the Bohr model is very well known to you. That is the 0.529, but they have given here 0.052 nanometer. Answer also in the nanometer, so we can keep it 0.052 n square by z. That is the hydrogen. So we are going to write here 1. And now you have to just keep the values here. 2 pi 3.14. This is the 0.052 and that is the 2. And you got your answer easily here. Yes, so this question, these two questions that I will say 5 and 6 are quite easy question. Question number 7. Actually in this paper you need to be selective that which questions are to do. Not every question is to do. Question number 7, you know that Brewster always we say that this is the fixed question, fixed data here. And in this one you know that mu is 1.73, that is the root 3. And you know that mu, whenever this is the root 3, this angle is going to be your 60 degree. And under the Brewster conditions, your reflected light is fully polarized. Partial is means refracted, the partially polarized. If you read the option, reflected light is partially polarized, wrong here. Reflected and transmitted, perfectly wrong here. Transmitted light, okay, is complete angle of, wrong here. And only you got the fourth option, reflected light is completely polarized and angle of incidence is 60 degree. So again, this was the easy question to do.
So till now we got some easy, some tough questions. Let's move further. Question number 8, one of the famous question, sometimes asked in the exam, but little different. They have given you stopping distance. You already know stopping distance will be equal to u square by 2a. You already know stopping distance, u square by 2a. But they're not talking about u and a. They're asking you about kinetic energy. They're asking you about force. So in order to generate S given, but U not given, A not given, they are talking in the terms of kinetic energy and they are talking in the terms of force. So in order to get force, multiply with M, divide with M. Now if you see from your S will become mu square by 2. This is your kinetic energy, that is K. M into A will be your force. So force on the bodies will become K by S. They are asking you FA by FB. So FA by FB will become KA by SA by KB by SB. And if you put from there, you'll get your answer K by KB into SB by SA. All the values are given, just put there, you'll get your answer. So not so easy. If they ask you only stopping distance, then it was easy, but it became little tough because they ask you to relate with kinetic. But it's not like that you have never seen such model. Such model already you got, only difference kinetic energy. They related, from there you can get the answer. Let's move further. Samayam ledu. We don't have more time. Question number 9. A wire is cut into 8 parts. So each part will have resistance R by 8. Now 4 parts you connected in parallel. So each part means 2 parts you have drawn, 4 you have connected in parallel. So this section, they are saying you 2 sets. So one set will have resistance R by 8 by 4. Other set will also have resistance R by 8 by 4. That is R by 32, R by 32. And now these two are connected in series, means your R net will become R by 16. This is not the question coming for the first time, previously also asked in the exam, but maybe in the examination hall, time pressure, you may have done mistake. And you have not only done mistake, many people have done this mistake. So don't worry, let's move further.
So now here comes question number 10, and in this question, you have seen this question in your class notes all the time. But here there is the one tricky part. In this one, if you see, they have given that this is your pressure drops to 11 atm. And this is the gauge pressure. And whenever we are using this PV equality and RT, we consider absolute pressure. So they are saying that we got here oxygen cylinder, and in this one some, okay, your moles are there, some gas is there. We are going to make some hole. And once we make the hole, some of the gas will leak out. And we need to figure out how much gas is going to leak out. That mass we have to find. They have given final pressure. This is the final gauge pressure is given, and you need to consider here absolute pressure. So we can write in this one, pressure becomes your 12 atm, volume is given here your 30 liter, and now we will use here PV equal to nRT. So you need to find whatever moles are left in your container. So we can write 12 into 10 power 5. Actually, you need to use 1.01 into 10 power 5 as they have given this data. Volume, you will write 30 into 10 power minus 3. These moles you have to figure out which are in the container are whatever given value. There is the 100 by 12. Temperature is 300 here. Now if you solve, this value comes close to 14.5. Here some people might have taken the gauge pressure. Your value comes around 13 point something. But we don't have to find this thing. We need to find how many moles leaked out. So you can say this is the 18.2 minus 14.5. Once you got this value, then you need to figure out the mass. So we can write m by mw equal to whatever this delta n value coming. And you can write here delta n into 32. Okay, so that will be your answer here. So even though this question is, yes, easy, but okay, we will mark this one as the tough because chances of mistakes are more because both answers are there in this one. Now, here comes one easy question from the optics. We were expecting the tough question will be there, at least one easy. They're saying 4 lenses are there, and you know that whenever 4 lenses like this, we are going to keep total power P plus P plus P plus P becomes 4p, and if you are finding the magnification, that becomes m into m into m into m, which is your m power 4. Okay, so this is the question that you see and you take the answer directly in the examination.
Let's move very quickly to question number 12. One of the famous model you have already done. Based on Kirchhoff's law, we'll start from, they're asking you VA minus VB means you have to start from A, you have to go to B, and in between you have to write the potential drop. So let's start from A. So VA, then going for inductor, you will write minus L into di by dt. Then for battery, you will write minus 5. Then for resistance, you will write I into R. That is 4. This will be equal to your VB. Now from here, you put the value. This is VA. If you put this value will come, di by dt is given 1, L is given 1. So this will come 1 minus 5 minus 4 equal to VB. Now from here, VA minus VB will come 10 volt. So not very tough question, but okay. For some people may be tough. Let's move further.
So here comes one easy question from the gravitation. Probably all people have done this question. They are asking in this one that what will be the value of g at this height, and you know here is your formula. Actually, they're asking the weight, so we can write in this one just mg. So we can write mg divided by this 1 plus h by r whole square, and this mg value at the surface of earth is given 48, divided by here 1 plus height r by 3, so we can write r and whole square, this cancel, this becomes 1 plus 1 by 3, which is the 4 by 3, so you can write 48 and 4 by 3 square, 16 by 9, and here becomes 27. So this one I will mark as the easy question. Now we got next question from semiconductor. So they have given this is the full wave rectifier, and in this one they are going to ask this at the t equal to 15 milliseconds. So first of all, we need to find at this time what cycle we are going to get here. So you will just put in this one 220 sine 100 pi, and this is the 15 into 10 power minus 3. You will get here 220 sine. This will be the 100. Okay, or directly now I can write, because you know this is going to be 15 pi by 10 means 3 pi by 220 sine 3 pi by 2, and you know this value is going to be your negative. So negative cycle is going to appear across this terminal. So you can write when we are having here negative, this is going to be your positive. Negative means this one is going to be your reverse bias, and here this one is going to be your forward bias. Now here there is one catch in this question, because NCRT is saying this thing, because they are not saying, but you know, in the transformer there is the phase reversal will happen for the pi. But as per the NCRT, because they have given one diagram for this one, so we are going for this one. Let's see. Okay, most probably, okay, if they are conceding the phase reversal, then answer slightly might be changed. Means phase reversal means here plus and here minus, but most likely answer will be given this one. Let's see what will happen.
भाई क्वेश्चन नंबर 15. Chalo bhai, two city X and Y are connected by a regular bus service. So let's say there's a city X. Now buses are coming out of the city. Let's say this is bus number 1. Let's say this is bus number 1. Now after time t, what they are saying, with a bus living in either direction every t minute. So what we can do here, ki after t minute, let's say bus number 1 is here. Then after that, bus number 2 will come out of this. Bus number 2 will come out of this. Okay? So tell me, if this bus is moving with a speed v, then how much distance 1 has moved when 2 has come out of the city? Distance will be vt. Forget about the unit, it will get cancelled out. Forget about the unit. Okay? Although they have said minute and we are taking v in kilometer per hour, but ignore it. Now after that, when 1 will go here, then 2 will be here, and then 3 will be here. The same thing is happening here also. If you take a city Y here, same thing is happening, bacha. Bus number 1 is here, then bus number 1 will go here, then 2 will be here, bacha. Okay, and so on. Now there is a scooty. ऐसा बनाने का scooty इधर. ठीक है. Now this scooty is moving from X to Y with how much speed? 60 km per hour. Now, if you take the reference here, okay, means with respect to scooty, if you solve, this is a question of relative motion. Then, with respect to scooty, if you see, or if you can say with respect to bus also, okay, so if you see with respect to bus, then all buses will be at rest. All buses will be at rest. Then what is the velocity of a scooty? So in one direction, it is in same direction, right? Same direction, relative velocity, we say minus. But in this case, if you see on this bus, with respect to this bus, so it will be in the opposite direction. So relative velocity of the scooty will be plus. So you can see this distance covered, relative displacement will be Vt divided by relative velocity. Relative velocity, same direction, we will write, let's say the bus velocity is more, V minus 60. And how much time coming? 30 minutes. Same direction it is coming, 30 minutes. Opposite direction, same distance, Vt. How much speed by? V plus 60. This is going to be 10. If you divide it, Vt will be cancelled, and when you solve, you will get V equal to 120 km per hour, and only one option is there, 120. And if you want to calculate, you better calculate. It's t equal to 15 minutes. Okay. Next question. Okay. Question number 16. Okay, classic question of conservation of angular momentum. We know this, we have done this. I1 omega 1 equal to I2 omega 2. You can write omega as 2 pi by t. 2 pi by t. So I1 you can take a solid sphere. 2 by 5 mr square divided by t1 equal to 2 by 5 m twice its present radius, 2r whole square divided by t2. So if you calculate, t2 is going to be 4 times t1. The t1 is 27 days, 108 days. Okay? Answer, sir.
Hello, Joe. Let's discuss the next question. This is standard model. This type of question have came in internal test also. This is present in GABE, ARMS, everywhere. So very standard question. Electromagnetic field in a plane electromagnetic wave is given by blah, 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 blah. This expression for the corresponding magnetic field, here subscript denote the direction of field. So kids, first of all, in these type of question, you must calculate the speed. The speed here given is standard 3 into 10 raised to the power of 8. Now we know that C has to be equals to E by B, right? E is given to us as 60. C we know from childhood, 3 into 10 raised to the power of 8 equals to 60 by B. So B comes out to be 60 by 3 into 10 raised to the power of 8. That comes out to be 20 into 10 raised to the power of minus 8, which is 2 into 10 raised to the power of minus 7. Now there are two options with 2 into 10 raised to the power of minus 7, option 1 and option 4. Now better remember that electric and magnetic field, they have the same phase. So whatever trigonometric function and phase you have here, same should be present there in answer as well. So that is also same. So now we have to work out on direction. We know that EB and V are correlated by this E cap, B cap, V cap. They have given me direction of velocity. Velocity direction is towards negative x because it is x plus vt format. So electric field is given in z direction. Velocity is given in minus I cap. So direction of B will be same as V cross I. V towards negative x, B towards E towards positive z. So negative x cross positive z. Direction of B comes out to be towards y. That's why the correct answer is option number 4. Let's move on to next one. Two identical conducting sphere A and B have their center separated by some distance. Charge of each sphere is Q and the force of repulsion between them is F. A third identical uncharged conducting sphere is brought in contact with sphere A first and then with sphere B and finally removed. You have to tell me what is the new force. So this is again standard question given in gym, given in grip. So now there are two spheres. Let's say both of them have by 2 charge Q and Q. So the force acting between them is KQ square by R square. Now you bring another non-charged conducting sphere of same radius close to first. So this will have charge 0, this has Q, total charge will become Q by 2, Q by 2. Now when you bring this same sphere to the second one, so just before touching, this has charge Q by 2 and this has charge Q. So total charge, which is 3Q by 2, will again get equally distributed. So this will get 3Q by 4, that will get 3Q by 4. Now once you remove this, now the charge on first one is half, second one is 3/4. So the net force, charge became half and 3/4. So the force become half into 3 by 4 times, which is 3 by 8 times. Hence the correct answer is option 3, which is 3F by 8. Moving on. This question, standard question, while teaching in class, every single teacher say dipole in uniform field. This question comes every year. So it is standard question, electric dipole of dipole moment this, aligned in the direction of uniform electric field of magnitude this. The dipole is rotated through an angle of 60 degree. The change in potential energies. We know that potential energy is minus P dot E, which is nothing but minus PE cos theta. Final angle is 60. Initial angle is 0. So when the final angle is 60, final potential energy will be equals to minus PE cos 60, and cos 60 is half. Initial potential energy when angle is 0 will be minus PE. Change in potential energy will be final minus initial. So what you get is minus PE by 2 minus PE. That simply becomes PE by 2. Now P is given, E is given. Simply multiply them. Very easy calculation, and then divide it with 2. You will get answer of 1 joule. Let's move on to next one. A microscope has objective of focal length A, eyepiece of focal length A. The total length is this. If the distance of the distinct vision for eye is this, the magnification of microscope is. So we know that magnification of a microscope can be written as LD by F naught FE. No doubt this is an approximate result. Length is given 40. D is given as 25. F naught is given as focal length of objective 2, and eyepiece has focal length of null. Very easy calculation. Same question came in our phase 12 examination. This is part of gym, this is part of grip. So it's not that physics paper was entirely tough. Physics paper is tough. Till now, all the questions I have discussed, they are all standard models, and they have been covered in the internal test, and they were present in grip as well as gym. So let's move on. The output Y of this logic gate implementation in the similar output as a dash gate. So better take here A, here B. This is NOR gate. So the output will be A plus B bar. And this second gate is here B, here A. The second gate is NAND gate. So what you get output from here is A dot B bar. Now both of them becomes the input. So this is A plus B bar. This is A dot B bar, and the output is an AND gate. So all you have to do is this dot this. Now, applying De Morgan's theorem, break the dot, change the sign. What you get here is A bar dot B bar dot A bar plus B bar, right? Now do term by term multiplication. So what do you get here? first becomes A bar dot B bar dot A bar plus second one betu A bar dot B bar dot B bar. A bar into A bar becomes A bar. B bar into B bar becomes B bar. So what you get is A bar dot B bar plus A bar dot B bar. And A bar dot B bar plus A bar dot B bar will be nothing but A bar dot B bar. Doing reverse of De Morvis theorem, we get A plus B bar, which is nothing but the output of a NOR gate. Hence, the answer to this question is NOR gate. Moving on to the next question. A uniform of rod of mass 20 kg and length 5 meter leans against a smooth vertical wall, making an angle 60 degree with it. The other end rests on a rough horizontal floor. The friction force that the floor exerts on the rod is. A standard question under the heading of torque balance. This question is present in gym. It is there in class notes. So this question is again standard question. In classroom, we ask what is the friction coefficient at lowest point minimum. But this question is present in everybody's notes. So you should have known this. Now here one thing, the option 1 is 100 root 3, not 10 root 3. So now here you have this rod. The rod have a length, let's say L. And L is given to us as, I think, 5 meter. The mass of the rod is 20 kg. And this angle with the vertical, with the wall is given as 60. So the angle with the ground is nothing but 30 degree by 2. Now, which forces will be acting on the rod? Mg, normal from the ground, normal from the wall, and friction. The normal from the wall and the friction force applied by ground has to have same magnitude because the net force in horizontal direction has to be 0. So all I am going to do is doing the torque balance about the lowest point to find the normal reaction from the wall, and whatever will be the normal reaction from the wall, same will be the friction force from the ground. So let's do it. Center of mass will be here at a distance of 2.5. Torque due to the force of gravity will be weight 200 into length perpendicular. Now this length perpendicular will be 2.5 cos 30. And cos 30 is nothing but root 3 by 2. So I am directly writing it root 3 by 2. This torque of weight about the lowest point get balanced by the counter torque of normal. Now how will you write the torque of normal? Extend the line of action of normal and find the length perpendicular. The length perpendicular comes out to be nothing but 5, the total length of rod, sine 30. So equals to n into length, which is 5, into sine 30, and sine 30 comes out to be half. 2 cancel. Here you have 5, there 200, cancel it out. 5, 200, you get 40. 40 into 2.5 becomes 100. So 100 root 3 is the normal. And same has to be the value of friction force. So if you have done gym properly, this question is present in gym. Now moving on. The current passing through the battery in the given circuit is, as soon as you see this question, you instantly realize this A, B, C, D, E, F, these all form a Wheatstone bridge. So here, this central guy, 6 ohm, is given just for designer purposes. So first of all, we are going to throw it out. Now, this 3 and 5 are in series. This 1.5 and 2.5 are in series. So this part resistance becomes 8, this part resistance is 4. So their parallel combination resistance will be 8 into 4 by 8 plus 4, which is 12. Cancel it out by 2. What you get is 8 by 3. So resistance of this part is 8 by 3. Resistance of this part is 1.5. Resistance of this part is 5.5. And see, they have given such a nice value. They have put internal resistance also as 1 by 3. So that this 8 by 3 and 1 by 3 becomes 9 by 3, which is 3. So what you get is 3 plus 1.5 plus 5.5. 5.5 plus 1.5 gives you 7. 7 plus 3 gives you 10. So the total resistance of the line is 10. And since the total resistance of the line is 10 and EMF is 5, what you get? Current is at 5 by 10, which is 0.5 ampere, which is option number 1. So now, this brings us to the next question. Now listen. This is a new type of question. A model for quantized motion of an electron in uniform magnetic field B states that the flux passing through the orbit of the electron is NH by E. What is NH by E? Flux passing through the orbit of electron. So we know that when a charged particle does circular motion in a uniform magnetic field, then the radius of circle is mv by qb. For an electron, we will write this q as the charge on electron E. Now they are saying that the flux passing through the orbit of the electron is NH by E. The flux passing will be magnetic field into the area of the circular loop. Area of circular loop will be nothing but pi r square. which I can write as pi into R, right? So magnetic, this is the total flux, and this is given equal to how much, Betu? NH by E. Now what I am going to do, I am going to pull a little bit of trick here. Rather than 1 of the R, I am going to substitute MV by EB. So we get B pi 1 R is there, and in place of 1 R, I am putting this MV by EB equals to NH by E. So darling, you can see BB cancel, EE cancel. This MVR part, this is the angular momentum. So the angular momentum comes out to be NH by pi. Beto, this is angular momentum. Though if you would have used directly the Bohr's second postulate, you would have used this only. Now what we are going to do is magnetic moment upon angular momentum equals to E by 2m. So now substitute in place of angular momentum NH by pi. So M upon NH by pi equals to E by 2m. Bring this NH by pi that side. So magnetic moment equals to E by 2m into NH by pi. Now they are asking for first Bohr orbit. So substitute N as okti. So what you get is Betu EH by 2 pi m, which is nothing but option number 1. Yes, this question I consider as a better question, and this question requires patience to solve. And in a lengthy paper, showing the patience for such a question is tough. So if you have done this question wrong, don't worry about it. But other questions which I have discussed till now, you should have done all of them right. So now moving on to the next question, Bachcho. Which of the following options represents the variation of photoelectric current with property of light shown on the x-axis? So we know that this is the standard graph. The photocurrent is directly proportional to the intensity of light. But this is kind of a trick question, because when you have got options like that, multiple things are correct. You try to make multiple things correct, but this is not what you have to do here. This option D is incorrect option. Reason being, here rather than having photoelectric current, if they would have written Ke max or stopping voltage, then this graph also would have been correct, but they have given photocurrent versus frequency of light. That's why this graph is incorrect, and the answer is A only. Okay, so next. Amit sir.
How about question number 26? देखा जाए. This is a question of velocity selector. We know this. What they are saying, an electron means charge moving with the speed whatever, injected to a magnetic field this much in perpendicular direction. So obviously it is going to perform a circular motion, right? But no, they are saying it should not deflect. So that means we have to cancel the magnetic force. Now Lorentz force, net force is given as QE vector plus Q into V cross B. If total force has to be 0, it gives us E vector equal to minus V cross B. Now what is the angle between V and B? If you take the magnitude, it becomes VB sine theta. Theta is 90 degree. So following that, the value of E is coming out to be C by 100. Velocity C by 100. We can say 3 into 10 to the power 8 divided by 100 10 to the power 6 into 9 into 10 to the power minus 4, which gives us 27 into 10 to the power 2. Okay? Now, if you follow this, E vector equal to minus V cross B, that means the cross product property is the cross product has to be perpendicular to the both vector. It means electric field has to be perpendicular to both velocity as well as magnetic field. So E is perpendicular to magnetic field. Also, the magnitude is 27 into 10 to the power 2. Question number 27. We will discuss at the last, because it is one of the easiest question they have asked. So we will discuss it at the last.
Hello. So question number 28, it is a very simple question. Here they have given the open organ pipe of frequency F. So fundamental frequency of open organ pipe, what we know? F is equal to what? V by 2L. Now they are saying the half of the length is immersed in the water. Then after immersing in the water, L by 2 length occupied. Now L by 2 is the air column length, and now what is the tube now? It is a closed organ pipe. Then what is the fundamental frequency? Suppose I am saying F closed, that is equal to what? V by 4, because closed organ pipe fundamental frequency 4L. What is the length of air column? L by 2. Now 2 cancel out, what we got? V by 2L, and V by 2L means it is a frequency F. This question we given in our face exam also. Okay. Then next question, question number 29. A parallel plate capacitor made of circular plate is being charged. Surface charge density on the plate is increasing at a constant rate. Now here surface charge is increasing, so electric field is also changing. Okay, when varying electric field is there between the plates, then displacement current will be there. And in the capacitor we have learned displacement current and conduction current will be equal. Okay, it means one thing, between the plates we are getting the magnetic current, that is the displacement current, so we will get the magnetic field due to the displacement current, and out of the plate we will get the magnetic field due to the conduction current. Okay, this all concept we know. Okay, so here what they are saying, the magnetic field arising due to the displacement current is constant between the plates and zero outside the plate. Magnetic field is not zero outside the plate, it is due to the conduction current outside. Okay, so that is wrong. Non-zero everywhere with maximum at the imaginary cylindrical surface connecting peripheries of the plate. If you are considering the plate like this, then we have done this question also in the class. We are taking the different radius circular area, and then we are getting like R by 2 when we are taking, then we are getting the different current. So when we are considering whole like this, then we are getting the maximum current, because all the current will pass through this plate or whatever cylinder we can say. Okay, so then we here what they are saying, non-zero everywhere, that is correct, because outside the plate it will be due to the conduction current, between the plate it will be due to the displacement current, with maximum at the imaginary cylindrical surface connecting periphery of the plate. Means you are taking here like this of the radius R we can say, then it will cover the whole electric field, so this displacement current will be maximum. Then answer is 2. Okay, 0 will not be, because what I said, outside it will be due to the conduction current, and between the plates it will be due to the displacement current. Okay. Then question number 30, it is a little bit lengthy, not difficult, because we have done this type of problem, but it is a lengthy 2 equations we have to make. And in so many time I said, when they are using the symbol, then what we will do, first we will assume in the mind R is equal to L by AK. So here they are using 2K, K, 2K, and all rods they given identical. Okay, then suppose you assume the R is equal to L by AK. Then what is the resistance of this? I can say R by 2, because 2 AK. Here what? R. Here what? R by 2. Now these 3 rods are in the series combination. Okay. If they are asking only junction temperature, one junction temperature, directly we marked up the formula also. Okay. But for the 3, what we will do? One thing we know here, in the steady state, rate of heat flow is a constant. Okay. So rate of heat flow means current. That heat flow I can say H is equal to what? Here and here, what is the temperature? 3T. Here, what is the temperature? T. What is the difference? 2T. Okay. When I am taking the end, like a current electricity, what we are doing, when we are taking the potential difference here, we will take the equivalent resistance. What is the equivalent resistance? R by 2 plus R by 2. So R equivalent, what we can say, R equivalent is the 2R. Okay. Then you will put here 2R. So 2 cancel out, H value, what we got? T by R. Okay, it is a rate of heat flow that will be constant, that will be equal in all the rods, because it is a series combination. Okay, now this we got. Now in between, like if I am taking T1 and T2, because I need the 2 equation for T1 and T2. Okay, because they are asking the ratio of T1 and T2. So here if I am writing the H value, then H value for the middle rod, what you will write? T1 minus T2 by, what is the resistance of the middle rod? R. Okay, and that H we got what? T by R. So I can equate with T by R. So R cancel out. So one equation we got in T1 and T2. T1 minus T2 is equal to T. Okay. Another equation we require to get the ratio. Another equation, anyone you can equate. Suppose you are equating here T2 minus T. If I am writing the H, T2 minus T upon R. What is R here? R by 2. So third rod I am writing, temperature difference is T2 minus T upon R by 2. 2 will go up. That is equal to, again I will equate with what? T by R. Okay. Now R cancel out. Another equation I will get from here. T2, 2T2 minus 2T is equal to T. From that you will get the T2. After getting the T2, put here in the first equation, you will get T1. Okay. Only equation making is little bit difficult, but all the concept is known for you. Okay.
So next question is of current electricity. Little tough, but okay, you can do, no problem. Yeah, okay. Question number 31. If you see here, these two are connected in parallel. So R net, if first you can find R net, then you can find the total current, how much coming, then you can think. So R net will come from here. These two are in parallel. They will give you R1, R2 by R1 plus R2. That will give you 3 by 4. And if you calculate from here, these two are also in parallel, R1, R2 by R1 plus R2, that will give you 8 by 6. That means it will give you 4 by 3. Now these two will be connected in series, means R net of the circuit will become 3 by 4 plus 4 by 3. That means from here you can calculate 12 and this will come I think 25. This R net, that means you can find current. Net current of the circuit will become V by R net. V is given 50. R net you got 25 by 12. Now from here you will get your net current. Net current will become 24 ampere. Means battery has given current of, means simple series and parallel. Simple series and parallel, then you have to do little bit, yeah. Means battery has given current, that current you already know, that current of the battery you got, that current is 24 ampere. Now this current is going to divide into two parts. You already know in parallel combination how current will divide. Inversely proportional, because these two are connected in parallel. So that's why we have to see how current will divide, yeah. So you already know in parallel current will be inversely proportional to resistance. So from there you can say here resistance is tripled. That means here if current will be X, here current will be 3X. Means total current will become 4X. 4X will be equal to 24, means X will be coming as 6 ampere. That means X value 6 ampere, means here current will become 3 into 6, that will become 18 ampere. And here it will become X is 6, so this will be your 6 ampere. Now here also you have parallel connection. Again same 24 current has to be coming for this. Here also current will divide. Again you will be having division of 24 in parallel. And now you can see these two are again connected in parallel. This resistance double, so current will be half. That means if this current is X dash, here current, this resistance is half of it. That means here current will be 2X dash. Now from here also total current in this part will also come 2X dash plus X dash, that will be your 3X dash. 3X dash will be equal to 24, means X dash will be coming as 8 ampere. Means here current coming is your 2X dash, means 2 times of 8. That means 16 ampere. That means you can see here your here current coming was 24 ampere. Yeah, that will be coming. Yeah, here current coming was 24 was total. Here current coming was your 18 ampere. But here current went only 16 ampere. Means 2 ampere is going where? Your Kirchhoff's current law, 18 coming, here 16 going. That means here current will be your 2 ampere. 18 coming, 16 going. So what is left? 2 ampere. 2 ampere will go from C to D. So that is what they are asking. Little tough question. No problem, no problem. Yeah, we can say tough question, but okay. Question number 32, again tough question for medical point of view. This is a tough question where you have to do differentiation. They have given you T, so T is given as X square plus X. So what you can do, first thing you can do is the differentiation. So you can differentiate both sides with T, and this X square differentiation will become 2X into dx by dt. X differentiation will become dx by dt. Now dt by dt, this will give you 1, and dx by dt is your velocity. So you can take dx by dt common. That means velocity you can take common. So velocity will be common. This will become 2x plus 1. That means from here you can get the function of velocity 1 by 2x plus 1. This is a tough question. This much of differentiation is not taught to medical student, but what we can do. Yeah, okay, no problem. Once you got this velocity, acceleration they are asking you, you already know whenever velocity depends on x, acceleration will come v dv by dx. Now V you already know, 1 by 2x plus 1. dV by dx you have to calculate. Again you have to differentiate. So 1 by x differentiation you already know, like that only differentiation will be there. So differentiation of this will become minus 1 by 2x plus 1 square into 2. Coefficient of x you are multiplying here, and if you check from here you will get your first option. Little tough. This question, both questions we can say question number 31, 32 are the tough question, but okay. Once exam completed, what we can do? We can only discuss the paper. So let's move further.
Now question number 33. Directly we have done the thermodynamics equation problem. It is a like same, only we have to use the area formula. Two gases A and B are filled at the same pressure in the separate cylinder with movable piston of the radius RA and RB respectively. On applying an equal amount of heat, so what they given? Equal amount of heat. Okay. And both the system reversibly under constant pressure. Pressure is constant. Okay. The piston of the gas A and B are the displaced by 16 centimeter and 9 centimeter. A means XA, I can say what? XA is the 16 centimeter and the XB is equal to what? Piston of the B displaced by 9 centimeter. This was a lengthy question. Maybe you don't have the habit. That is maybe the problem. Otherwise, question is not difficult. If the change in internal energy is the same, delta U also they given same. Delta UA is equal to delta UB, we can say. Heat same, QA is equal to QB. So heat same, delta U same. Then what we can say? Q is equal to delta U plus W, then W same. Okay, so work done we can say what? Same. So WA is equal to WB. Okay, work done is equal to now pressure they given constant, underline constant pressure. For constant pressure, work done is what? P delta V. So pressure constant, so both side pressure will get cancelled out. It means delta V we have to consider. Delta V means what? Area into displacement. How much extra is there? That is a derivation we have done. So area into displacement. So area, I can take area of the A, that is a pi RA square. Pi RA square into what is the displacement of piston A? 16. That is equal to what? Pi RB square into 9. Okay. Now pi cancel out RA by RB. RA by RB whole square, what you got? 9 by 16. By taking the root on both sides, you will get the 3:4. Okay. Question number 34, little bit difficult, that you cannot think in the exam directly. Okay, so here what they given, in an oscillating spring mass system, a spring is connected to a box filled with a sand. Like suppose you have taken this is a spring and it is a box, and in this sand is filled. Okay, and sand leaks out slowly out of the. Slowly, what is important here? Box vertically, so that the average frequency WT and average amplitude AT of the system changes with time. Which one of the following option? Change omega and the amplitude variation we have to show. So here what is happening? Slowly coming out means relative velocity, we can say what? 0. Means this sand is not applying any force on the block. Okay. Only means spring force and the gravity is there. So conservative forces are there. Okay. So when the sand is coming out, so for the spring mass system, we know that omega is equal to under root of K by M. Okay. Now what is happening? Mass, as the sand is going out, mass is decreasing, then what will happen? Omega will increase, and it is not a linear variation. Okay. So then we can say what? Omega is increasing. One thing clear. Now what about the amplitude? What will happen with the amplitude? Here it is coming out slowly. That is only the problem here. Maybe amplitude you have unable to decide. So here it is coming out slowly means force is 0. Force is 0 means at any point where whatever the velocity is there, that velocity will not get affected because only conservative forces are there, and due to the sand there is no force will act. Okay. So like maximum velocity is what? A omega. Now omega is increasing, then A will decrease. So what will happen? We can say amplitude will decrease and omega will increase. Not linearly. Okay, so now you can see the options here. Omega is increasing, amplitude is decreasing. No any other option is there like that, then directly we can go to the answer 1. Okay.
Oh yeah, question number 35. Moment of inertia question. Let me solve it on the note. Ah, bhai. So they are saying, just a minute. There is a sphere of radius 2r. From here, another sphere of radius r has been taken out, correct? It has been taken out like this. Now they are asking if you calculate moment of inertia about this diameter, about this y-axis, then you have to take the ratio of moment of inertia of this removed part, this smaller part. Let's call this moment of inertia I1 about this axis. Now this y-axis is acting like a tangent, है ना? It is a tangent to this smaller sphere. Okay? Now what we can write, bacha? If you write the mass of the smaller sphere, let's call it m1. It's nothing but density into 4 by 3, let me tell you, this is 2r, so its radius will be r, 4 by 3 pi r cube, correct? Density into volume. The total mass is density into 4 by 3 pi 2r whole cube, right? Total mass. Now, first of all, we will try to calculate moment of inertia of the smaller, I1. So I1 will be simply about the tangent. If you apply parallel axis theorem, it will be 7 by 5 m1 into r square, which you will get 7 by 5 into rho into 4 by 3 pi r cube into r square. Okay? This is I1. This is the moment of inertia of the smaller one. Find the moment of inertia of the complete object without any cavity. So I2 will be, now for the complete sphere, this is just a diameter. So you don't have to use 7 by 5 mr square. You have to use only 2 by 5. But I will use m2 into 2r whole square. M2 is how much by? So it's rho into 4 by 3 into pi 8r cube and then multiplied with 4r square. Now the moment of inertia of the remaining portion will be I1 minus, I2 minus I1 divided by what they are asking. Just a minute. Now they are asking it, take the ratio of I remaining and I1. I1 is, we have calculated this I1 over here. Let's say this is 1, this one is 2. Here whatever we got, this will be 3. Now they are asking, take the ratio of I1 divided by I remaining. If you do the calculation, I think the answer is 7 by 57. The calculation I am now doing here. Okay. Then the next question is, plates of a parallel plate capacitor separated by distance d. Now two slabs of different dielectric constant K1 and K2. Thicknesses are 3 by 8d and d by 2. Means if you add, please understand, if you add 3d by 8 plus d by 2, it will come out to be 7d by 8. Means the remaining area is vacant. There is air. So that area will be, distance will be d minus 7d by 8, which is d by 8. So the diagram will be like this. There is nothing there. Then there is K1 and then we have, let's say K2. This is d by 2. This is 3d by 8 and this one is k equal to 1 air. This is d by 8. Now you can consider it like a 3 dielectric connected in series. So what is the k equivalent? Formula, it's T1 plus T2 plus T3 divided by k equivalent T1 by K1 plus T2 by K2 plus T3 by K3. Now T1 plus T2 plus T3 is nothing but D. How much is K equivalent? So if you see here, they are saying that the capacitance become 2 times. The capacitance become 2 times, means if you remove all these and replace only one dielectric, then the capacitance become 2 times, means K equivalent is 2. Again, these three addition will be D and K equivalent is 2 because they are saying the capacitance becomes double. Now after that, put the value of T1. T1 is D by 8. The value of K1 is 1. T2 is 3D by 8. And the value of this K1 here, the value of K1 is 1.25. 1.25 you can write 5 by 4. Correct? So here I will divide with 5 by 4 times K2 plus D by 2 into the value of K2 I will write here K2. Okay? If you solve this, calculate K2 from here, then you can write K1 equal to 5 by 4 into K2, the answer you will get is 2.66. Again, the calculation, it's a lengthy paper. Okay, calculation please, you do bacha party. After that, question number 37. Question number 37 is a standard result. You know this. If there is an inclined plane, smooth inclined plane, a block is going down, angle theta, then here time taken to go down is t naught. Initially it is starting from rest. You take similar inclined plane, similar but rough mu, then block is taking a time n times. Obviously here time taken will be more because it is rough surface, so time taken will be more. So it is n times. So here n they are saying bacha, it's 2 times. So our n value is 2. Now the value of mu in this case, we will not do the derivation. Mu equal to tan theta 1 minus 1 by n square. The value of theta is 45 degree. So it will be 1. 1 minus 1 by 2 square will be 4. 3 by 4. This calculation I will do. Okay. Next, question number 38. Again a tough question because it's lengthy. See, in a question of vertical circular motion, always take care of two things. First, you have to apply circular motion dynamics equation. Means something you will do mv square by r, and the second thing you have to apply conservation of mechanical energy. So here they are saying ki the string gets slack at some point p. So if you draw a free body diagram, only mg is acting, its component here mg sine theta is the centripetal force. So if you write it, let's say the velocity here is v, so it's mv square by L. So v square we are getting Lg sine theta. V square is Lg sine theta. Now number 2, equation number 2, conservation of mechanical energy directly you can apply like this. If you find the height here, this length, so this is theta, so this will be theta, this is L, so this height will become L sine theta. Okay? Here velocity we are saying it is v. I have told you the lower velocity square equal to higher velocity square. Obviously it is more because lowest point it is more, it is less, so we will do minus 2gh. H is from here to here, so L plus L sine theta. Okay, this is equation number 2. Now what you can do, you can put here v square equal to Lg sine theta here, and then calculate v naught square. It will become 2gl plus 3gl sine theta. This is our equation number 3. What they're asking v divided by v naught. So if you divide 1 and 3, Lg will get cancel. You will get sine theta divided by 2 plus 3 sine theta. But understand, this is v square, v naught square, where we are finding v by v naught. So we will take under root. So the correct answer is option number 3. Question number 39. Manish sir.
Here they given the two container. One has the volume 2 liter and other has the volume 3 liter separated by the partition and thermally insulator. Thermally insulator means heat will not go out, so total energy will remain constant. Okay. In one they have given the mole 5, in other they given mole 4. Pressure also they given. Okay. This type of question we have done when the temperature they given, mole they given. So we are equating the internal energy. We are taking the total internal energy is constant. By that we got the formula of the equilibrium temperature and all. Okay. But here they are not talking about the temperature. They given the both side same gas, that they have given gas, not gases. They said the gas means we have to consider both sides same gas, but they have the different number of moles, different pressure. Okay. So what we will do now, temperature they have not given, so we will think the formula of the internal energy in which pressure and volume will come. So that you mugged up F by 2 PV. Okay. So we can take what? One container has the pressure what? 1 atm. Okay. And degree of freedom same gas. Suppose I am taking that is a F. So F by 2 P1 V1. P1 V1 plus F by 2 P2 V2. This is the internal energy before when they were separated. Okay. When you remove this, then what will happen? Now it is removed. Then finally it will come to the common pressure. Okay. Then common pressure. But same gas or degree of freedom will not get affected. That is F by 2. Suppose final common pressure is what? P. And now they occupy one container. So total volume, what you will take? V1 plus V2. V1 plus V2. Now put all these values. This F by 2 cancel out. P1 V1 means here 1. Here V1 is what? 2. Plus here F by 2 also cancel out. P2 what they given? 2. V2 what they given? 3 is equal to what? P, and V1 plus V2 what you will get? 2 plus 3, that is a 5. After solving easily you will get the answer. Okay.
So this is again one question done so many times. They are asking you RMS current. You already know IRMS will be coming as VRMS by Z. Z you can write under root of R square plus. Your XL is more, so XL minus XC whole square. Now from here, if you put the values, all the values are given, you will get the answer. And they're also asking you phase difference. So tan phi you already know, tan phi you already know. That will be coming as XL minus XC by R. So from here, these two formula, all values are given. Just put, you will get the answer. Let's move further.
In 41, they given the radius of the Martian orbit around the Sun is around 4 times the radius of the orbit of the Mercury. Then they given the time period of the revolution, that is a 687. For the Mars they given, and they are asking the time period for the Mercury. Okay, so that is based on the for two different planets. It is based on the Kepler's third law. T square is directly proportional to R cube. Then T, suppose I am saying they are asking for Mercury. So okay, upon T Mars, that is equal to what? T square. Suppose I am using the square also. R2 means R Mercury upon R Mars whole cube. Okay. Now what the data they given? Time period of Mars they given what? 687. Okay. Take the root on both sides. So T Mercury they are asking. T Mars we know that 687. That is equal to what? Radius of the Mercury. Here they are saying the Martian here is this. And radius of the Martian orbit around the Sun is around 4 times of the Mercury. So means this is the 4 times. So this ratio what you will get 1:4. So 1:4 whole to the power 3 by 2. Why we are taking the root on both sides? Here is square here. Okay. Now here what we are getting 1/4. We can take the 1/2 square. 1/2 square means it will come 1/2 cube. That is 1/8. Then T ME what you will get? T ME is equal to 687 by 8. And you are very perfect in the mathematics. That's why you are checking the exact answer. But here exact answer will not come. Okay, so approximation you have to take 8 6 8. Okay. Now you can solve. Okay. This gravitation question is lengthy, but if you just focus on this, whatever they given here, they given the quantities, normal symbol they use like density rho, radius r, surface tension s. So here one thing they given, value of the 4 or 5 variables they are asking, a, alpha, beta, gamma, delta. So one relation they given what? T directly proportional to S to the power alpha, A to the power beta. So what we will do, from this we can write one equation for the dimension in the terms of dimension. T is the time period. So LHD, what we can say, LHD is equal to L. Okay. Then RHD, what we can say, RHD is the surface tension dimension is what? MT minus 2. So MT minus 2 whole to the power alpha. Okay. A is the what? Area they given. So area is what? L square to the power beta. Then they given the rho. Rho is the density. Density is what? Mass upon volume is ml minus 3 whole to the power gamma. And R, what they given? R the radius. Radius means L to the power delta. Okay. Now from this what we can say? M to the power alpha, and here you will get the gamma. Okay. L power what we are getting? L to the power 2 beta minus 3 gamma plus delta. Okay. And what is the T power? T power we are getting minus 2 alpha only. Okay. Left hand dimension and right hand dimension equal. LHD is equal to T. Sorry. Time period they given. LHD is the T. Now here, if you are equating to T to the power minus 2 alpha and left hand T to the power 1, so we can say what? Minus 2 alpha is equal to 1. So alpha is equal to minus 1 by 2. One variable you got. Okay. And no need to find all 5 variables. They are not asking how you got the answer. Okay. So alpha is equal to minus 1 by 2. Now any other we can take where the only 2 variable we are getting, like alpha plus gamma. Alpha plus gamma is equal to what? Right hand side alpha plus gamma, mass dimension, and left hand side what is the dimension of mass? 0. Then we can say what? 0. Then what we got? Gamma is equal to minus alpha, and alpha we already solved minus 1 by 2. So gamma what we got? 1 by 2. Check it. Maybe it is a like no any option that is contain the alpha minus 1 by 2 and gamma. Only one option it is there. So here, alpha minus 1 by 2, gamma minus 1 by 2, not possible. Alpha minus 1 by 2, gamma 1 by 2. Here alpha 1 by 2, not possible. And one more. Here alpha 1 by 2. So only one option is possible. What? Alpha minus 1 by 2, gamma 1 by 2. No need to find the other. Everybody got or not? Okay. Then question number 43 we asked in the test also. Here a particle of mass m is moving around the origin with a constant force F pulling it towards the origin. Bohr model is used. Now here they said F force is acting. Okay. So I said what? We have to use always two equations. One, whatever the force acting on the particle, that we have to equate with centripetal, and one Bohr quantization principle. Okay. So what we can say here, F is equal to what? mv square by r. Okay. And one thing they given, force is constant. Force is constant, and this constant, then we can say what? V square is directly proportional to R, or V directly proportional to R to the power 1 by 2. Okay. That we got from the force equation. Second, Bohr quantization. Bohr quantization is what? MVR is equal to NH upon 2 pi. Okay. Now, here R and V we have to find. V relation already we got. V I can replace by under root R because I need proportionality. Okay. So V I can replace by root R. So this you will get R to the power 3 by 2. H upon 2 pi is constant. So it is directly proportional to N. Now take the reverse root. So R you will get N to the power 2 by 3. So R directly proportional to N to the power 2 by 3. Then V directly proportional to R to the power 1 by 2. 1 by 2 when you will do, then what you will get? V directly proportional to N to the power 1 by 3. Okay. Similar question, same data we asked in our PACE exam. Then question number 44. Two identical point masses P and Q suspend from the two separate massless spring of the spring constant K1 and K2. Means KP they given, K1, and KQ they given what? K2. Okay. Oscillate vertically. If their maximum speed are the same question we already asked, the ratio AQ by AP. They are asking the AQ by AP of the amplitude. So AQ by AP they are asking. And they are saying that their maximum speed are same. What is the formula of the maximum velocity? A omega. So AP into omega P, that is equal to AQ into omega Q. Now AQ by AP, what you will get? AQ by AP is equal to omega P by omega Q. Here they are asking what? Now omega we got here. Omega P by omega Q. Omega formula is what? Under root of omega is equal to what? Root over K by M. Mass are same, so omega directly proportional to root K. Okay, so omega P by omega K we can replace by under root of Kp by Kq, and Kp and K1 by K2 it will come. Okay.
So final question, question number 45, whose direct formula also you know, so directly use it. So you already know once the ball strike the ground and rebounded back, impulse on the ball directly you can write m into under root of 2g h1 plus under root of 2g h2. All the values are given, mass value given, h1, h2 given. When you are putting these values, this is finally giving you 21 newtons. A direct formula, every standard formula, everyone remembered. So you will get answer 4. So that's how we have completed our paper discussion of physics. What will be cut off? How was the paper? How many easy question? How many tough question? That will be discussed tomorrow. We cannot discuss in hurry. Discussing in hurry will make unnecessary mistakes. So that's why for that we will have meeting tomorrow. But as per our knowledge till now, entire India, everyone is feeling physics to be very tough. Toughest paper in the history of Neet or AIPMT. So that's why cutoff will decrease. 617 is a very good mark. Someone is a 617. Thank God you did not ask 720. How is 720? Thank God. Thank God. Yeah. This is like putting salt on the wound. Some injury happens, someone is putting. So yeah, that's very good. Karthik, you have rocked the NEET exam. Don't worry. Yeah. SC cutoff, ST cutoff, BCA, BCD, all the cutoffs will be told tomorrow in a separate meeting in the detailed manner. So don't worry. Till now we are expecting 720 only. Someone is asking, sir, what is the highest marks in Gosalites? We always expect 720. Let's see. That also will get to know tomorrow. That also will get to know tomorrow what will be the highest marks of the Gosalites. Till then, relax. Exam over. So much of hard work done. Now relax. Don't take any tension. All the things we'll discuss tomorrow. Ta-ta, bye, bye, bye. One question is left, which will be discussed by, if you're interested, you can stay. That question everyone left. But our Amit sir is saying I will discuss, so he will discuss question number 27.
Okay, last 5 minutes. The question number 27, actually it is a kind of revenge question. Last year 720, 67 students were there. So they just want no one get 720. So the last question there, it's a J advanced level, not also mains level. Okay, it's J advanced level. So don't worry if you have not done it. Okay, we will discuss it. I will explain you the question there. On this side there is water, it means liquid is there. This side there is a wall, right? So obviously due to surface tension, this side will be pulled up, just like in a capillary rise you might have seen it, right? Now, if you let me explain here. See, let's say there is a wall here. So the tangent, it is making an angle theta naught at this point, right? Tangent, it is making an angle theta naught. So please understand this, in capillary rise also what we have discussed, from this reference level, whatever is the weight of the liquid is there, it has to be managed by the force of the surface tension. Okay? So what is the force of the surface tension? Please understand this, Bachawadi. In 3D, if you understand this dimension they are saying it is Z. Any dimension, they have not mentioned it. Take this dimension in the back side 3D, you have to imagine, take it like Z. Now they are asking what is the curvature, what is the equation of this? I don't know, it's not a straight line, obviously it's not a straight line, it is not going to y equal to mx plus c. Definitely something parabola, I don't know. Okay, so let's find it. So what we will do, I'm taking the origin here, you can take it here also, it doesn't matter. So I will take any point at a distance x. Let's say the height of the point is y. So I'm only interested in this portion. From here to here, only this portion I'm interested, okay? Now if you visualize it in 3D, give me 2 minutes. In 3D, it is just like a wedge, but obviously it's not an inclined plane, it's a curvature, right? So this dimension, it is Z, this is Y, This is X, this length I don't know. But you have to understand this side liquid is there. Yeah? This side liquid, this is behaving like a stretched membrane. So this side liquid will pull it. The surface tension force will be in this direction. How much? So total how much length is connected? Z. So it will be S into Z. Similarly here also. It will be S into Z. Let's say tangent at this point is making an angle theta. This one was theta not given. Let's say this angle is theta. So this angle is theta. Now this will become SZ cos theta. Don't worry about the horizontal side. This side pressure will be there. So this SZ cos theta plus remaining forces will balance out in horizontal direction. What about vertical direction? So the mg of this wedge will be balanced by SZ sin theta. Here comes the mathematics. SZ sin theta equal to mg. Now, as they said, sin theta in the question, they have said you can take approximately equal to dy by dx. Mass, we can write density into volume. Oh, density into volume of the wedge. How to write volume of the wedge, sir? So what I will say, if this area, the front side area is A and the depth is Z, I'm eligible to write A into Z into Z. Now, sir, how to write area? We don't know area. This is not a triangle. How can we write area? Let me tell you. First of all, Z cancelled. dy by dx equal to rho g. How to write area? Arey bhai, area is nothing but, area under a curve is nothing but integration y dx. Integration y dx. Okay? Now, sir, here integration, here differentiation, what to do? So what I will do, I will differentiate it once more. Here I am integrating from 0 to y because I am interested only in this portion, not the entire portion. So if you differentiate it again, it will come double differentiation. What is the differentiation of integration? So bhai, you are integrating y, although what I am going to say is not completely true, but understand, if you are integrating y, then if you differentiate the result, you will get back y only, na yaar. By putting the limit from 0 to y. Now if you solve this, you will get rho g y by s. Now you think yourself, is this question belong to Neet? Obviously not. So it's a revenge question. Don't worry if you have not done it. Okay, bacha buddy? Take care. Tata. Bye-bye.
Stay tuned for tomorrow meeting. Your all doubts will be cleared. Tata, bye-bye. And come on, yeah.